Okay, good morning, everyone. I'm really happy to host you for the fifth time for the World Cancer Day. The actual date is Tuesday, but we did it today so everyone can be here or online globally. This is a podcast globally and also recorded. So I would like to welcome all of you. I'm really grateful to Merck and Teva for supporting the Cancer Biology Research Center meeting for the fifth time. They have been traditionally sponsoring this meeting, so I'm really grateful. Thank you. Some key cancer facts, which will sound very grim, but we're here today to remember that there is lots of innovative science going on, so to keep some optimism. There are 10 million people dying of cancer each year, about 20 million new cases of cancer in a year. We know that 30 to 50% of all cancers are preventable, about 10% are heritable, and 40% of cancer-related death are linked to modifiable risk factors. We all know this picture. It hardly changes, but in the last 2 years, it changed a bit. If we're looking at pancreatic cancer, for example, for female being the third cause of death from all cancers, and prostate and breast remaining the estimated new cases at the top line, lung as the second, and colorectal as the third. And the same goes for male, just changing the prostate and lung for the estimated cases of death yearly. We know that the statistics for 2025 are that more than 1 in 3 people will develop cancer during their lifetime. The incidence rises in women and decreases in men, interestingly. Pancreatic cancer is rising, incidence and mortality, so I'm happy that some of the speakers will speak about pancreatic cancer today and new hopes for diagnosis and therapy. The new cases for both sexes, taking all ages from 0 to 85, are about 20 million in 2022, while in 2050 will rise to 35 million. And the death, accordingly, will double by 2050 from cancer. So we have a lot of work to do, but already in 2024, cancer remained the dominant focus of drug developers, with 15, which are 30% of all drugs approved, novel approvals in 2024, and this includes even several first-in-class approval, as well as the 11th PD-1, PD-L1. So one may wonder why we need 11 PD-1, PD-L1 blockers, but some of the speakers will talk about immunotherapy and specifically about this target, this axis as a target. We see that research continues to advance immunotherapy. We have some cases. I will not follow on it, but you'll have this presentation available. It continues to harness the power of molecularly targeted therapies on both immunotherapy and targeted therapy. We'll hear a lot about today. And we have an amazing lineup of speakers. I will leave you with the beginning. I just want to invite Professor Dan Pe'er, the VP R&D of Tel Aviv University, who is hosting this venue again for the 5th time. So welcome.
Okay, thank you, Ronit. It's become a tradition already, the 5th time. And before I'm going to thank everyone, I just want to say that as we have heard, cancer remains the second cause of mortality and worldwide. So we are obligated to deepen our understanding in the cause of cancer, improve our prevention, diagnosis, and of course therapeutics for cancer. And today we'll hear, looking at the program, outstanding speakers from Israel in different fields, different areas. And you can see the diversity of investigators at a very high level of science coming from, as I mentioned, diverse field in order to tackle one disease, which is probably 500 different types of diseases from different angles and different aspects. So with that, I just want to say thank you for Merck and Teva for sponsoring this for a very long time, the fifth time, Ronit, Professor Ronit Sachi-Fainaro, and Dr. Judith Ben-Porat, who is really the driving force of these days in Tel Aviv University and I think among Israel. And I wish you a very successful event, eye-opener and optimistic, although we've already heard that we'll double the time, the people who will die from cancer in 2050, but I hope that with this huge optimism and novel approach for therapeutics, diagnostic, and prevention, we will be able to really put an end in cancer. Some places even delete the word cancer, okay? In some institutions, some of you may know what I'm talking about. I wish you really an intriguing event, and always ask questions because you have stellar scientists here, and this is a great opportunity also for graduate students, postdocs, to really ask the best and the brightest in this field right now. Ask questions, very important. Thank you, and I wish you great success.
Thank you, Danny. As this is broadcasted, so I ask you, the speakers, to remain on time, not to use a laser pointer, but the mouse, because we need everybody to be able to see it on the screen on their computers. It's also recorded, I remind you. And I encourage questions from the audience, short questions. I will repeat every question so it can be heard online as well. So without further ado, I'm really happy to invite Professor Yossi Shilo from Tel Aviv University, the Faculty of Medical and Health Sciences, to speak about cell senescence and genome instability, emerging insights, and the new player.
Thank you, Ronit, and thanks to the organizers for inviting me to speak here today. As you know, our view of cancer is through the prism of genome instability, and that's because we are interested in a genome instability syndrome, AT. And one of the aspects, the cellular aspects of AT, is premature cellular senescence. And what I will try to do today is to convince you that understanding cellular senescence is extremely relevant to today's meeting. So AT is a multisystem disease which involves, as you can see here, the cerebellum, which degenerates, the immune system, many other tissues in the human body, and it's a genome instability syndrome, like many others, all of which are caused by severe defects in the repair of DNA lesions, and the acute sensitivity to ionizing radiation that you see here in the bottom actually reflects a specific defect in dealing with a specific DNA lesions, double-strand breaks. But in addition, please note the cancer predisposition. Here's the cancer relevance and the premature aging, which together with the genome instability highlight a very solid axis connecting genome instability, premature aging, and cancer predisposition, all represented in this disease. AT is caused by a total lack of the ATM protein kinase, Exactly 30 years ago, beginning of 1995, the corresponding gene was identified in our lab, and we and others later found that ATM is actually a very powerful protein kinase, and you can see here at the bottom its role in maintaining genome stability. The DSB stands for double-strand breaks, but in addition, you see many other cellular circuits, metabolic pathways, and also triggers of this powerful kinase. So, in fact, this is a homeostatic protein kinase with numerous functions, numerous roles, involvement in many cellular pathways, all of which are presumably missing in patients with AT. And we've been studying the physiological and molecular basis of the various symptoms of AT, trying to link them to specific functions of ATM, asking, okay, so what is out of the many functions of ATM, what is the specific function whose loss is responsible for this or that symptom? And today I will start with the premature aging, which actually caught my attention when I started to work on AT in the late 70s as a grad student. And I could see when we visited the homes of AT patients that some of them in their early 20s looked very, very old. Since then, this phenotype has been substantiated. And as you know, part of aging is the increase in the amount of senescent cells in various tissues. And so senescence is relevant here. And in fact, it could be seen already in the early days of AT research as premature senescence of primary cells derived from the patients. You probably all know about cellular senescence, but I decided to show you this introductory slide because our view as newcomers to this field is slightly different from the traditional one. So we think this is a dynamic cell state and not a terminal cell fate. Okay, this is a huge distinction. And of course, it is expressed as extended cell cycle arrest, but with potential reversion to cycling, what we call senescence escape. Now, I know some of you might ask me, are you sure that within this population of senescent cells, there are no cells that actually are not senescent? But the fact is that this population of senescent cells can stay like this for weeks, months, and even years in our body. And there's no cell cycling, no cell division there. And they show many of the hallmarks of cell senescence, which is never defined by one hallmark, always a combination. And then all of a sudden, there is cell division. And that's what we call senescence escape. It is very, very relevant and important in the big field of senescence to today's discussions. So there are profound alterations, of course, in cell morphology, metabolism, chromatin organization, and these cells are resistant to apoptosis. So they are not even close to death, cell death. They are very, very alive, and they metabolize very actively. That is also very, very relevant. Please bear this in mind. And other than the replicative senescence that you're all familiar with, you know, the Hayflick limit, senescence can be induced by a variety of triggers, for example, genotoxic stress. And by saying genotoxic stress, I deliberately and purposely include ionizing radiation, which is used in cancer therapy, and the entire array of chemotherapeutic drugs. Each and every one of them has been shown to induce not only cell death, but also cell senescence. And there's a whole range of other stimulators of senescence. A major phenotype of senescent cells, as you know, is the senescence-associated secretory phenotype, which is basically active secretion of a whole array of bioactive molecules, which actually causes sometimes paracrine effects And senescent cells can affect not only the microenvironment, but also have some systemic effect. But this process has like Janus phases, because on the one hand, it is important and plays a role in normal processes, development, tissue repair, tumor suppression. And on the other hand, it plays a role in aging, degenerative, and inflammatory processes, and it can accelerate the aging of tissues and also cancer. Like I said, it is characterized by many readouts. This is one of the common ones, the one that everybody checks, which is activation of the senescence-associated beta-galactosidase. But again, senescence is always defined by a combination of several readouts. And during aging, it has been known for a while, the proportion of senescent cells increases in various tissues. And again, it's not necessarily a one-way street. So this is important in therapy-induced senescence. This is a formal term, TIS, because, think about it, senescent cells, senescence can be caused by chemotherapeutic drugs and ionizing radiation. And the population of these tumor-derived senescent cells can stay in some niche in our body for weeks, months, and even years during the apparent remission, and sometimes even the patient can be declared as cured. So what happens when there is senescence escape? This could potentially be one of the mechanisms of tumor recurrence, sometimes years after the appearance of the primary tumors. So going back to AT, so what brought us to this field? The cellular phenotype of AT is characterized by chromosomal instability, radiation sensitivity. We knew that all along. I did a lot of work on this as a grad student. But at that time, the cells that we were working on, which were primary fibroblasts derived from skin biopsies, were notorious for poor growth in culture. They didn't grow well. And the reason was that in most cases, laboratories got them from cell banks or other labs at advanced passage level. But we raised them, actually, I raised them from freshly taken skin biopsies. And they grew beautifully until at relatively early passage, they stopped growing, they became flat. You know what it is. We knew at that time what it was, although we didn't have so many readouts for senescence. So I developed myself one of them, which is actually decrease in colony-forming ability, which appears long before the senescence is shown in tissue culture. And so we knew it was senescence, and that was, I think, the first characterization of this aspect of the cellular phenotype of AT. And, you know, they are radiosensitive, but if you don't irradiate them, they're fine. But this is something that happens without any exogenous trigger. They just senesce prematurely. And we thought that it could be linked to the premature aging of the patients. So you can see here on the right-hand side that paper from a while ago, and a paper we published just 2 years ago, which is actually part B of the same story. This paper begins where that paper ends. And this project was led in the lab by Majd, who just graduated, got his PhD and left the lab, and with the very, very active daily involvement of Yael, Dr. Yael Ziv, who is here, our lab manager. And you can see here also our collaboration with the groups of Rani Elkon in our department and Dudi Tzfati in Jerusalem, with a group of very enthusiastic students. And we also collaborated here with Judy Campisi, one of the pioneers in the senescence field, who sadly passed away a year ago. So what Majd found was that the initial question was, what happens if we lower the oxygen pressure in the incubator to the physiologic level? And what Madge found is that it indeed extends considerably the proliferative lifespan of AT cells, but they still senesce prematurely. And when he did the transcriptional analysis, he could see that basically the question was, so what kind of senescence is it? Is it simply accelerated replicative senescence? And the answer is yes, with some variations, but basically what we see here is what I saw many years ago, accelerated replicative senescence. And they also exhibit the SAS, so it's a typical senescence. And what we could see also is robust activation of the interference, a whole group of interferon-stimulated genes, which are main drivers of cellular senescence. And the data suggested that the active pathway here is the CGAS-STING pathway, which is activated by cytoplasmic DNA. And in this case, cytoplasmic DNA can emanate from the micronuclei, which we often see in these cells, which are genomically unstable. These micronuclei finally erupt and spill their DNA into the cytoplasm. And this system, which originally evolved to deal with exogenous DNA from viruses, is activated. And you can see here the typical activation in this diagram of the CGAS enzyme, which synthesizes the dinucleotide CGAMP, which in turn activates STING, the stimulator of interferon genes, which in turn recruits very powerful kinases that phosphorylate transcription factors that finally lead to the expression of interferon and the downstream interferon stimulated genes. So that was the thinking at that time. And then much further developed this work, and now using cells derived from the animal model, the mouse model of AT, and these are ATM deficient mice. We usually prefer not to work with MEFs. So here is an example of cells from adult mice. These are primary lung fibroblasts, which can be very easily established and turn out to be a wonderful tool to study this. And this also resonates with another symptom of AT, which is, we think, is related to the premature senescence of cells in vivo, and that's the interstitial lung disease, which is the second cause of death of AT patients after cancer. So this work appeared a few weeks ago, and you can see here other the involvement of other people in the lab, of course, Yael and Jan, who's here, and I'll discuss Jan's work in a minute. Also, a wonderful collaboration with three groups in our department, Uri Ben David, Karmit, who will speak here today, and Rani Elkon, again with Dudi Tzfati. And in here, what Majd found was that if you just use a regular incubator, these cells, the wild-type cells, grow like MEFs and senesce very quickly. But if you go down to the physiologic oxygen concentration, wild-type cells never senesce. They keep growing, and eventually they are immortalized, and they also acquire malignant potential. Without any treatment, without any, you don't touch them, you just grow them under 3%. So this is a valuable system to follow these cells all the way from being innocent primary fibroblasts to tumors without any treatment at all. So what happens with AT cells, or actually ATM deficient cells? These are mouse cells. They don't grow at all under 21%. They do grow under 3%, and they senesce very quickly. So the difference between wild type and ATM deficient cells is now much more dramatic compared to the human healthy and AT cells. And to cut long story short, what Majd found was that indeed the main axis of this leading to this very rapid senescence within 40 days is the CGA-STING pathway, but there are additional players here. For example, P38 and P53. So if you inhibit chemically either CGAS or P38, or you delete P53, you can evade this senescence. And the transcriptomic analysis, again, was very similar to what we saw in the human AT cells. And one interesting aspect of this study is that we could use a chemical inhibitor of ATM applied to wild-type cells. Remember, these cells grow forever, okay? And this caused senescence within 10 days, okay? But this is a reversible inhibition, and so when we removed the inhibitors, the cells that were in deep senescence that we could even show by transcriptomics was identical to that of ATM deficient cells. They came out of senescence, and within 2 weeks, they were growing happily. So again, this is a system where you can induce senescence and release from senescence very easily. So senescence became a subject by itself in the lab, so kind of an offshoot of our studies of AT. So from now on, I won't mention AT anymore. And the person who is actually leading the senescence-only studies in the lab is Jan, who is here. Jan is about to submit his PhD thesis next month. Jan came to us from France on a Marie Curie fellowship, and I'll show you part of his work. So one of the characteristics of senescent cells is ongoing DNA damage. If you just use any readout of DNA damage, you don't treat them with anything. You don't do anything to them. You see ongoing DNA damage. No wonder, because they are very active metabolically, and metabolism is accompanied by the ongoing generation of reactive oxygen species, which are the primary generator of DNA damage in our body as we speak. But why we don't suffer so much from this? Because there are DNA repair mechanisms that are working all the time to remove these lesions. So we would expect them to work harder in these cells. But Jan's attention was caught by sporadic reports on attenuation of specific DNA repair mechanisms in senescent cells. This is counterintuitive. And the question that he thought about, and he came to me with it, was, is this a general phenomenon? And I should again say that Jan has 100% of the credit for conceiving this project, thinking about it, carrying it out, and finally writing it. Of course, with the daily involvement of Yael, and I was trying just not to interfere, but it was his idea. So what is general meaning? Does this apply to most or all DNA repair mechanisms, various senescence inducers, all kinds of senescence? If so, could this become a senescence hallmark? So Jan was thinking cell biology, and I was thinking cancer biology. Again, thinking about therapy-induced senescence. What does that mean if this is true for therapy-induced senescence? That these cells sitting in our body, senescent, quiet, not dividing, are actually accumulating a lot of DNA lesions. And if they go back to cycling, within one replication cycle, all of these lesions with which their DNA is decorated become mutations, chromosomal breaks, which could explain the aggressive therapy-resistant nature of recurrent tumors, as we all sadly know. So this work was also published just last month, and you can see in the title of this paper and the graphical abstract that this indeed was the conclusion. This was quite a complex bioinformatic pipeline that Jan built, spanning 60 transcriptomic data set. And most important, he didn't approach the supplementary data of the paper. He took the accession numbers and went to the databases and took out the raw data. which is very important because it allowed him to do uniform pre-processing, which is a very important stage here of the raw data in his hands. And that allowed accurate data set comparison and even integration. So you can integrate various data sets and compare growing, proliferating versus senescent cells and see strong phenomena. And you can see this in this simple heat map where blue is repression and this beige-brown is activation. And to cut long story short, among the repressed genes, and you can see here also replication senescence, stress-induced premature senescence, and oncogene-induced senescence, various types of senescence, various cell lines, and the pathways that are marked with the red arrows are all the major DNA repair pathways suppressed across the entire study. Now, you might have wondered about the unusual color code here. This is not what you usually see in papers. It's derived from this wonderful famous drawing of Edouard Manet. You see, these are exactly the same colors. They go throughout the paper. And when you do gene set and enrichment analysis, now this is according to specific repair pathways. You see the same phenomenon. Here again, these heat maps now divided to different repair pathways. And no wonder the protein products of these genes often interact. Now, when you in this map of the various repair pathways, of course, I won't get into the details, the blue boxes represent the suppressed proteins or the suppressed genes or protein products are shown here. And you can appreciate that in each and every pathway, there is sufficient number of key proteins that are suppressed to inactivate that pathway altogether. And importantly, this does not happen in quiescence. This is specific to senescence. So if you group together genes whose expression pattern is similar, and you do this in primary cell lines now, not under stress-induced senescence, but simply undergoing replicative senescence. So these are the fibroblast primary cell lines that you grow in the lab, and they advance in passage levels until they hit senescence. And you can see these patterns of various forms of decrease, all of them containing DNA repair genes. Here in the heat map, you can see the sharp decrease towards the overt senescence. And there are some studies who did proteomics. You see the same phenomenon, again, representing DNA repair proteins, and see the similarity between the gene clusters. And it's quite amazing. I realized it when I prepared this slide, and these are proteins. And not much time is left, so I'll just mention that Jan did experimental validation. This is double-strand break repair. This is UV damage repair. This is oxidative stress caused damage. And in all cases, in senescent cells, you see a marked decrease in the repair dynamics. And you also see this decline now in fibroblasts derived from people at different ages. So here you see different ages at which these biopsies were taken. And again, in these clusters, you see nice representation of DNA repair. When you look at the overlap of the genes reduced at aging or in senescence, overlap is here in the middle. You see there all the major DNA repair groups. So this actually the connection between senescence and aging, and this is the final conclusion. You can see here the decline of DNA repair and the elevation in DNA lesions, which obviously mean a lot when these cells go out of senescence. And indeed, what's expected after therapy-induced senescence is this extreme genome instability, therapy resistance, and increased aggressiveness of these tumors. And I hope I convince you that these are indeed time bombs. And I guess all of you are thinking, okay, why not do what's called 1-2 punch therapy? First treat with chemo or radiation, and then with senolytic drugs to get rid of these senescent cells, right? But there are a lot of caveats, the heterogeneity of senescence, the timing, and most important, remember, senescence, if you do a systemic treatment with senolytic drugs, you may also interfere with normal processes. So this is not very good. And I'll just end with this collective picture of our current group. I don't know if there is time for questions, but I'll be happy to answer during the coffee break.
Thank you. Thank you. Can you hear me? Thank you, Yossi. Wait 1 second. And a bit of to embarrass you. Congratulations on amazing talk and congratulations on your birthday. I think we have
No way to keep secrets these days.
a little bird told me. We have time for one short question from the audience. Any burning question? Maybe? Yes, Karmit.
Karmit asks a question, you have to brace.
Short. Do you see differences in, so if DNA damage repair is a cause, okay, of senescence, so do you see differences between different tissues? So some tissues that are more exposed to DNA damage, so the load of DNA damage is higher, so skin or lung, compared to other tissues, or maybe you can then define the DNA repair ability of each tissue? So aging of the different tissues differently?
Okay, this study was mainly done in cell lines, but actually your question touches upon a very basic topic or subject in DNA repair, because obviously our skin is exposed to the most abundant carcinogen in the world, which is sunshine. But our deep tissues are the DNA in our deep tissues is exposed most of the time not to exogenous agents, but rather the enemy from within, which is reactive oxygen species. So, most of the reviews or even papers in their introduction, they start with DNA damage is caused by radiations and chemicals and so forth. My way of doing it is DNA damage is caused by reactive oxygen species, and occasionally by external DNA damaging agents. So the answer is, as far as deep tissues are concerned, that's the kind of damage we all have to deal with, including patients with genome instability disease. And it's important because this is how we have to look at it. AT patients are not irradiated, okay? They suffer from, or their DNA suffers from the same spectrum of DNA lesions that we all have to, we all encounter. So, and it is true for all patients with all genome instability syndromes. Question is how they deal with what we call the everyday DNA damage.
Prerogative of the chair. Quick question. Xenomorphic or senolytic drugs are more relevant here?
That's a very good question. But again, I mean, my worry, my concern is that if this is not directed, and it's no way to direct these drugs to the mysterious niches in which these islands of senescent cells exist, any systemic treatment with such drugs might be problematic. And this has been suggested years ago, this two-punch, but we don't see anything yet in the field.
Thank you. Our next speaker is Professor Ruth Schertzoval from the Weizmann Institute of Science, who will speak about tumor-stroma coevolution. Thank you.
Okay, thanks. Thank you, Onit, for the kind invitation. And thank you all for coming today. And I'm happy and excited to tell you a little bit about the work that we do in my lab, studying tumor stroma coevolution and mostly cancer-associated fibroblasts in the tumor microenvironment. So we view tumors as ecosystems in which the cancer cells that we heard a lot about this morning play very important roles, but they cannot progress or metastasize without support of other cells in the tumor microenvironment, and these include various types of cells, such as immune cells that we hear a lot about, especially with the development of immunotherapies, but also the cells that we are interested in the lab, which are the fibroblasts or cancer-associated fibroblasts, or CAFs in short, which I'll use today. And fibroblasts are, or CAFs are the most abundant cell types in many different tumors, many aggressive tumors such as pancreatic cancer, and they perform a variety of pro-tumorigenic activities, some of which are described here. They are traditionally known for their role in matrix remodeling or in wound healing, but in recent years it became very clear that they serve in many other functions, such as regulation of of the immune microenvironment. They have different metabolic effects. They secrete different factors that recruit other cells from the microenvironment. And like other cells of the tumor microenvironment, CAFs are considered to be genomically stable. So their pro-tumorigenic activities are not driven by mutations to the DNA, but by something else. And we are interested in understanding this something else. And we know also today that CAFs are highly heterogeneous, both in function and in their transcriptional programs. So some of this heterogeneity is described here, and CAFs perform different activities that we'll talk about more in this talk. But they're also diverse in their origins. So different origins have been proposed for cancer-associated fibroblasts, including tissue-resident fibroblasts, but also adipocytes, parasites, mesothelial cells, and others. So the development of single-cell technologies allowed us and others several years back to start to decipher this heterogeneity in the transcriptional level. We did this in a mouse model of triple-negative breast cancer, and we identified two main populations of CAFs that we termed at the time PCAF and SCAF for expression of protoplanin and S100A4. And we found that these populations are actually composed of different subpopulations that change as tumors progress and metastasize. So in the PCAFs, we see here the normal mammary fibroblasts, and as tumors metastasize and progress, we go through inflammatory fibroblasts, wound healing fibroblasts, ECM remodeling fibroblasts, and so on. And in the SCAFs, we found this population that we termed at the time antigen-presenting CAFs, because they expressed MHC class II. And these three populations, the myofibroblastic, the immune regulatory, and these MHC class II expressing CAFs, have emerged in other tumors and other tissues. And we find them, at least the myofibroblastic and the immune regulatory CAFs, in most carcinomas that we look at. The antigen-presenting CAFs are a little bit newer. And I'd like to also mention another subset that has recently emerged, and we look at it a lot in the lab now, which are the cancer-associated parasites that have been gaining more and more interest. So in our view, the overarching challenge is to understand how do normal tissues turn into cancer-promoting environments, or how do normal fibroblasts turn into cancer-associated fibroblasts, and what are the factors that reshape normal tissues into distinct microenvironments? So how will one microenvironment be different than another microenvironment? And as we heard in the introduction this morning, cancer is actually a lot of different diseases, so how is the tumor microenvironment different in different diseases? And our hypothesis is that it's a combination of mutation dependencies and organ dependencies that contributes to the formation of different tumor microenvironments. So different mutations in the cancer cells will lead to different rewiring of the microenvironment around them, and also different organs with their structures and their functions will dictate different microenvironments to similar mutations. And we study this question in a variety of models, looking at patient samples in collaboration with clinicians, mouse models, and cell co-cultures. We do ex vivo functional profiling. We use fact sorting and analysis, different modalities of imaging, laser capture microdissection, and RNA sequencing. And we focus on three main types of cancer: colon cancer, pancreatic cancer, and breast cancer, but we've also been looking a little bit at gastric cancer and recently at ovarian and prostate cancer. And the mutation that we've been focusing on most, and I'll tell you more about today, are actually two mutations in BRCA1 and BRCA2 genes. And in organ dependencies, we've been focusing mostly at stress responses, but I'll not talk about that today for lack of time. So if you remember the PCAF and SCAF that I described earlier, that was in a mouse model. When we looked at those markers in patient samples and breast cancer patient samples, we found that actually the ratio between protoplanin positive and S100A4 positive CAFs correlates with disease outcomes. So the SCAFs are good, so to say. The PCAFs are bad, and if you have more PCAFs than SCAFs, the survival is worse. And this is in triple-negative breast cancer, and since this is an Israeli cohort, this was in collaboration with Einav Galyam and Maya Dadiani at Tel Shomer, we had information about BRCA mutations in these patients. And when we looked and stratified the patients according to the BRCA mutations, we found that, in fact, this SCAF to PCAF ratio and the correlation with survival is true for the BRCA mutated patients, but not really for the BRCA wild type. So this was sort of the first study to associate the CAF compositions with BRCA mutations. That this was a correlation, and it suggests that maybe the cancer mutations affect the fibroblasts, but we also have this mutation in every cell in the body of these patients, as there's one copy of this mutation. So we wanted to look at it more functionally, and also we asked whether this is specific to breast cancer or more global. And so we moved to another BRCA mutated cancer, and this is pancreatic cancer that Talia Golan here works a lot on, and she's also a collaborator on this project. And in collaboration with Talia, as well as with Memorial Sloan Kettering, we looked at these CAF subpopulations in a cohort of BRCA mutated and wild-type pancreatic cancer patients, and looking at the three main CAF subsets using smooth muscle actin, clustrin, and MHC class II for the three different subsets, and excluding cancer cells and immune cells. By multiplex immunofluorescence, we found that there is a clustrin positive subpopulation that is elevated. You can see it here in red in BRCA mutated patients. And so this is still a correlation. In order to show that this is really caused by the cancer mutations, we used mouse models and ex vivo cell co-cultures where we injected cancer cells that we knocked out BRCA2 from them, so to mimic the BRCA mutation. And so the cancer cells don't have BRCA, but all the cells in the mouse express wild-type BRCA, and we could show, and I'm showing here just the schematics of this because this has already been published, that in fact when we do these procedures, the CAFs become more immune regulatory, suggesting that it's the BRCA mutations in the cancer cells that affect the fibroblasts to become more immune regulatory and less myofibroblastic. So this study showed that it's really the BRCA mutations in the cancer cells that affect the fibroblasts. I didn't show you that this is driven through a transcriptional regulator called HECHOK factor 1, which we are now trying to target pharmaceutically in the lab in order to see the effect, and we have very promising super preliminary results that I won't show today. But again, BRCA mutated cancers are not just in breast and pancreas, so we wanted to look at the two other BRCA mutated cancers, and we're doing this now, looking at ovarian cancer and pancreatic cancer, to see whether the same mutation in different organs leads to a similar or different microenvironment. And I'll just show you, this is preliminary, but still I had to show some unpublished data. So this is preliminary work in ovarian cancer, where again in collaboration with clinicians at Memorial Sloan Kettering, we've been looking at BRCA mutated and wild-type ovarian cancer patients, and this is looking at pentachrom staining that stains for what you see here, so we can look at different parts of the ECM. And what we find is that BRCA wild-type patients have more collagen, whereas BRCA mutated patients have more mucin. So they have different stromal compositions. We also looked at published data set still from MSK of ovarian cancer patients, and we single-cell RNA sequencing, and we looked at their stromal compositions. So interestingly, in ovarian cancer, this immune regulatory calf population that I described earlier is very small. Instead, what we see are mesothelial cells, and those perform, at least transcriptionally, immune-related functions. And I won't get into the details, but we also see similar stromal changes in BRCA-mutated patients in this cohort. So and we're currently looking at prostate cancer, but what it seems from looking at all these cancers, the picture that is emerging is that BRCA mutations shift the stromal microenvironment from myofibroblastic to immune regulatory. And this, of course, has clinical and therapeutic implications. All right, so in the last few minutes of my talk, how long do I have? 5, great. I'd like to spend a few minutes talking about this immune regulatory function of the cancer-associated fibroblasts. And so CAFs are supposed to block the tumors from forming, but in the case of cancer, we know that they're pro-tumorigenic and they actually block immune functions in various different ways. A lot is known about their interactions with T cells. Less is known about interactions with other cell types, and we wanted to take a sort of unbiased approach to look at the interactions of CAFs. And so we did this computationally by looking at single-cell RNA sequencing data from breast cancer patients and looking at ligand receptor interactions. This is the work of Shimrit, a former PhD student in my lab, in collaboration with Bar-Ilan's lab. And what we did here was to ask, who do cells interact with? And what you see here is what we knew before, that CAFs are the most important cells in the tumor microenvironment, but now we actually have proof for that they are at least the most interacting cell type in the microenvironment. And this is shown here. And they interact with various cell types, but the cells they interact most with is tumor-associated macrophages. And we then applied network motif analysis, and it's exciting to have Shai here in the audience, wherever Shai is, who started this in Uri's lab. And what we did here is looking for motifs in the network of the tumor microenvironment. And again, we see that CAFs are the most interacting cell type. They interact mostly with macrophages down here, sending signals to them, but also to other cells in the microenvironment. But we wanted to look at cells that they don't necessarily interact with and ask, why do they not interact with them? And so we asked, do CAFs modulate NK cell activity? And NK cells, natural killer cells, are supposed to be killing cells that invade or harm the body, but they don't really do this in cancer. And so we wondered if CAFs had anything to do with it, and we took an approach of isolating cells and mixing them ex vivo so that we can manipulate them in different ways. And when we do this with NK cells, cancer cells, and CAFs, we find that CAFs inhibit the ability of NK cells to kill cancer cells. So this is shown here. Normal fibroblasts don't inhibit the cytotoxicity of NK cells, but these PCAFs that I showed you before actually do. How is this mediated? So we looked at receptors for activating and inhibitory pathways in NK cells, and we found that 2 of the main activating receptors, NKG2D and DNAM1, are inhibited in the presence of cancer-associated fibroblasts. what about the ligands? Are those expressed on CAFs? So the answer is yes. So we find that CAFs express ligands for NKG2D and DNAM. And when we block the activity of these receptors on the NK cells, we inhibit cytotoxicity. And this is mediated through, through CAFs. And I'm not going into this in order to finish on time. But finally, when we asked, is this relevant to the human disease, we went back to patient samples. And again, in collaboration with Einav Galiam, we stained the same cohort of triple-negative breast cancer patients for different ligands for the NK receptors. And we find that the expression of Nectin-2 on cancer-associated fibroblasts, one of these ligands, is correlated with poor survival. So to summarize this work, what we find, or what we suggest, is that CAFs serve as decoys for NK cell receptors, so they attract them. The NK cells kill the CAFs instead of killing the cancer cells, and by this, they are exhausted and cannot kill the cancer cells. And this was the work of Aviad, a former postdoc in my lab, together with Yael, a current postdoc in my lab, and Koral, who was a PhD student in my lab and is here in the audience. All right, so to summarize everything I showed you today, CAFs originate from different sources and diverge into various subpopulations with distinct tasks. And so if I showed you this figure before with the three subsets, today we think that, and we know that it's more complicated, and that the subsets are not discrete. And when we want to look at this in different organs and in different contexts, we actually need to look at all the markers, because some of these CAFs can transition from one to another, and some share markers, and we should actually look at their markers and their functions. So the tumor microenvironments, we believe, are shaped by cancer mutations and organ dependencies, where the adaptation to organ-specific microenvironments requires distinct crosstalks, and the organ-specific microenvironments instruct common themes to different cancer types. So finally, this is my group in our last, the year before, Hanukkah party. And these are our collaborators, and thank you, and I'm happy to take questions.
Thank you, Ruti, for this wonderful talk. We have time for 2 questions from the audience. Yes, Carmi.
Do you think some of the fibroblasts migrate with the tumor from the primary sites to the next site? So then you have a mixed population of fibroblasts, but some are resident and some are coming with them? Like
Yeah, that's a great question. So if we think that the fibroblasts are migrating with cancer cells to the metastatic site, that's a great question. We don't see evidence for that, and we think that it's mostly that in the new site there's a new microenvironment that is formed. Some of it may be pre-formed before the cancer cells arrive, but I don't think this has really been resolved. The expert for the metastatic CAFs will be Neta, who will join us later. Neta Erez here, who is my great and close collaborator as well.
Hi, wonderful talk.
Thank you.
A question about the BRCA2 story. So you think there's a direct interaction between the cancer cells and the CAFs, or this is mediated by other types of cells?
Yeah, so I didn't get into the details. We did look at the secretome of these cancer cells with and without BRCA, and we see differences, and we see molecules that could be targets or should be targets of HSF1. So we think that it could be directly mediated by what the cancer cells secrete. We don't know that it's necessarily BRCA or the DNA damage caused by BRCA, because we didn't see differences between BRCA1 and BRCA2 in pancreatic cancer and in breast cancer. We do actually see differences in ovarian cancer. So we think that, yes, that it is direct interactions between the cancer cells and the fibroblasts, also because of the in vitro experiments, but we also know that the cancer cells with BRCA mutations can affect immune cells. So there's probably also effects that are through that.
Thank you, Ruti.
Thank you.
The next speaker, as we transfer from basic to translational to clinical talks, I'm happy to invite Boaz Nachmias from Hadassah Medical Center, who will speak about advances in acute myeloid leukemia, clinical and scientific insights. Thank you.
Hi, good morning, everyone. Thank you for the opportunity to come and speak today. I'm sorry I'm not at the scope of the previous speakers, but I'll try to give you a little bit of clinical perspective and show you how the research we are doing is actually translated to the benefit of the patient. So I'm the head of the leukemia service at Hadassah Medical Center, and I have a lab studying leukemia stem cells. And just a brief description about acute myeloid leukemia. Is the most common and fatal leukemia in adults. And basically, the bone marrow tissue, which consists of hematopoietic stem cells, that generates most of the cells in our blood. The leukemia stem cells are hematopoietic stem cells that cannot differentiate it properly and proliferate and basically consume the bone marrow tissue, eventually leading to anemia, thrombocytopenia, and neutropenia, which cause the main symptoms of the patients, which fatigue, infections, and tendency to bleed. And if we look at survival of patients with acute myeloid leukemia with AML, we see that they have a pretty dismal prognosis. In elderly patients above the age of 60, we see a very low survival at the 5-year mark. And even in the more younger patient population, we see not very good survival. And the advantages you see over the years is mainly due to supportive care and improvement in transplant. And the main treatments that we are treating AML today is not very, very different from the ones we treated about 40 years ago, and are basically our chemotherapy and then either more chemotherapy or going to allogeneic bone marrow transplant. And not to say that there weren't any developments over the past year. I think if I can mention a few major leaps, is one of a better understanding of the molecular profile of AML, which mutations drive the leukemia, and trying to target these mutations. And this is just a small slide to give you a sense of that. For example, FLT3 mutation, this is a tyrosine kinase that generates signals for proliferation and survival. So we have FLT3 inhibitors that are incorporated either in first line or in relapse patients, and other inhibitors that we use. But these agents did not really revolutionize the field. They're added to the backbones most of chemotherapy. But I think one of the true revolution in the fields in the past, I think 10 years, is the combination of azacitidine and venetoclax. Azacitidine is a hypomethylating agent, and venetoclax is a BCL2 inhibitor. And this combination is very effective with roughly 50% CR rate, complete remission rate. And the main thing is it's low toxic. So we can give this combination in elderly and non-fit patients and achieve high rate in remission. And at the age of 80 or 90, this is very, very significant. When I started my internship, we didn't have much to do with these very elderly patients, and now I'm giving them this combination and achieve cure in a lot of patients and give them a really good quality of life. The main issue about this combination that it's not curative. Patient will go into remission but will not be cured, and almost all of the patients will eventually relapse. And we now understand better which patient will have durable remissions and which will not. So we know that, for example, patients with IDH2 or NPM1 mutations will probably be more sensitive to this combination and achieve long-term remissions, and patients with RAS activation will not. And in the lab, we're really trying to understand better how to attack leukemia and mitigate relapse. And if we think about leukemia, we can think about the leukemia stem cells as the root of the plant, and that generates the bulk leukemia. And when we try to attack the leukemia stem cells, we try to look at driver mutations that really initiate the leukemia, such as the NPM1. I discussed it briefly. Co-mutations or co-activations just promote leukemia, and actually what is the fuel of the leukemia, what is the metabolomics that drives the leukemia. I won't touch on that today. So just an example on how we translate the clinical insight into the lab. So as I mentioned, NPM1 patients usually have favorable prognosis. This patient has long-term remissions, but we identified that a subset of patients have a very late relapse. And that's an enigma in itself, right? Because you think about a proliferative cancer, why does a patient goes into remission for a year and then a year later the disease comes back? I would expect the turnabout time would be much faster. So we looked at all of these patients that have the NPM1 mutations and have a late relapse, and we did sequence to look about these mutations. And surprisingly, we identified that most of the patients with late relapse have atypical NPM1 mutations. And I'm not sure you are aware of that, but the typical NPM1 mutation actually changed the localization of NPM1 from the nucleoli to the cytoplasm. But these patients with the atypical mutations and the late relapse have aberrant localization of the NPM1. It's not in the nucleoli as the wild type. It's not in the cytoplasm as the typical mutation. It actually stucks in the nucleus. And so that really brought us to try and understand how is the change in the localization of the atypical mutation promotes the late relapse. And at this time point, we did an assay that we are doing routinely in the lab, which is BioID. This is basically a proximity ligation assay to try and map the interaction of a specific protein. In this assay, we take the protein of interest, we connect it to a BIR-A, which does biotinylation to all the proteins in the close proximity, and then we do a streptavidin-based pull down and send to mass spec analysis. So we get a map of the protein interactors of our specific protein of interest. And just one slide about that, we see a very different interactome, maybe not surprisingly, because the typical NPM1 sits in the nucleoli, the atypical that makes the high rate of relapse sits in the nucleus, and the typical mutation sits in the cytoplasm. And we see a different interactome of the three mutated proteins. And just a taste, one of the most significant interactors of the atypical mutation in the nucleus is a protein which is called RREB1. It relates to RAS signaling, and our hypothesis, and we are currently validating that, is that perhaps the fact that the atypical NPM1 stacks in the nucleus, it promotes RAS signaling, and by that it generates a more resistant disease. Another study in the lab that's carried out by one of my PhD students, Vika. Here we again try to look at critical vulnerabilities in leukemia and leukemia stem cells. So we started by a CRISPR screen of 30,000 genes in AML, and obviously we identified a lot of genes. Some of them are just housekeeping genes or unrelated genes that won't be targets for future therapies. So to try and narrow the list down, we took this roughly 600 genes that were critical for the viability of AML cells by our CRISPR screen, and we looked at two different data sets. One is which genes are upregulated in leukemia stem cells. As I mentioned before, the leukemic stem cells is the root of the leukemia, and these are the cells that generates the relapse. And the other thing is we looked at genes that are upregulated in relapse. And when we took these three lists together, genes that are important in our CRISPR screen for the viability of AML cells, genes that are upregulated in leukemia stem cells, and genes that are upregulated in relapse, we had just one protein that was shared by all three lists, and that protein is IPO11. It was a protein that was not known for its significance in leukemia, and we did some work about, we silenced IPO11, we saw that it reduced proliferation of AML cells, reduced differentiation, and reduced engraftment in mouse of leukemia stem cells, both from patients and also patients with relapse disease. And IPO11 is one of the proteins of the importing exporting family. I'm sure you know about CREM1, the major exporting protein, but there are also canonical importing, and IPO11 is one of the proteins that import stuff into the nucleus. And again, we turn to the BioID. That will be a repeated motif in the talk today. And we did BioID for IPO11 to try and understand what protein it takes into the nucleus, and one of these proteins we identified UBE2E3. We validated that with Western blot to see that once we silence IPO11, you can see it here, UBE2E3 does not appear in the nucleus. And we did also a lot of work about UBE2E3 showing that it basically recapitulates the phenotype of IPO11 knockdown. So we think that IPO11 mediates the nuclear entrance of UBE2E3 into the nucleus, and by that it promotes leukemia. So again, this is work with RNA-seq of UBE2E3 knockdown cells. So we see that once we knock down UBE2E3, we see the cells are becoming less leukemia, less stem-like, more differentiated in several data sets of leukemia stem cells and the TCGA data sets, meaning that UBE2E3 supports stemness or supports leukemia stem cells. And this is the last time we did BioID for UBE2E3, and this was a really remarkable results. We saw that we got in the BioID almost all of the interactors, all of the proteins of the polycomb repression complex, and specifically polycomb repression complex 1.1. Now, PRC is a complex of protein that affects gene translation. It's critical for hematopoiesis and differentiation, and specifically PRC 1.1 is a complex of regulatory proteins that is responsible was shown to be significant in AML. So we got all of the interactors of the PRC 1.1, such as BCOR, Ring 1, KDM2B, in our CRISPR screen. So we believe that UBE2E3 is interacting with PRC 1.1, and indeed, once we do a knockdown of PRC 1.1, we see a decrease in, once we do a knockdown of UBE2E3, we see a decrease in PRC 1.1 activity. So just in summary of this project, IPO11 is a very essential gene for the viability of AML cells and leukemia stem cells. We know that IPO11 mediates the nuclear localization of UBE2E3, and UBE2E3 supports stem cell gene signature through positive regulation of the non-canonical PRC 1.1. And obviously now we are trying in the lab to see how we can target either IPO11 or UBE2E3 as novel targets, as novel therapy approach. My team and collaborators, just to finish, so Ayat, my lab manager, and Vika is the student who I showed you all of the work about UBE2E3, Liran from Weizmann, and Aaron Schimmer from Princess Margaret, where I did my post, and obviously my mentor, Professor Dina Ben Yehuda, and Professor Moshe Gat.
Thank you, Boaz, for the beautiful example from bedside to the bench and fully back. Questions? She's going to Karmit already. Yes, Michael.
Very interesting talk. What do you predict would be the effect of UBE11 targeting of normal hematopoietic stem cells, which are very similar to leukemia stem cells?
That's a good question, obviously. We know that IP11 affects also normal hematopoiesis. We're not sure currently what will be the effect, but I think that always when we think about targets in cancer, we're always worried about the effects on normal cells, but history shows that at the end of the day, the cancer have some dependencies that are makes it more vulnerable to certain targeted therapy. Like if you think about the inhibition of CREM1, which is the canonical exporter of proteins from the nucleus to the cytoplasm, you would say that would be a very bad target. Probably you will have a lot of off-targets effect and a lot of toxicity. Still, this is a drug that is used today in myeloma and AML, and so there are many examples of that. So basically, we need to really push forward and see what is the toxicity in cells that are not related to the cancer, and hopefully the toxicity won't be too much.
Thank you. I think we'll leave time for questions during the break. Mm-hmm. Our next speaker is Karmit Levy from Tel Aviv University, who will speak on melanosomes' role in melanoma immunity. Now someone else has to ask questions.
I'll ask myself. Hi, good morning. Thank you so much. Thank you, Ronit and Yudit, for putting it up, organizing it, supporting students all along these years, allowing them to travel and allowing us to do some science. It's amazing. It's really, really amazing, and thank you for this CBRC. Okay, so now I'm going to tell you a story that some of you heard. Most of it will be unpublished, and I will go talk about something related to your wonderful talk, Ruti. So let's start. This is a pathology of melanoma. You can appreciate here the in situ melanoma. It's a skin cancer, as you know. I marked here kind of the melanoma in situ. You can see that melanoma is still concentrated in the epidermis of the skin. This is the dermis, the lower part. And you can appreciate the brownish color. Okay, so if you imagine from the outside of the skin, this looks like a huge and atypical nevi when you look. But this is brownish. It is indicative of the origin of melanoma. And melanoma is originated from melanocytes. Melanocytes are the cells of our body located in the basal layer of the skin. And they are producing pigment which spread it to the outside of the epidermis, okay, generating the color. We have a great paper with Shai Shen-Or when we established a model. He established a mathematical model of this skin pigmentation. And so these are the melanosomes. Melanosomes are these extracellular vesicles. They are 5 times bigger than exosomes, secreted from those melanocytes, and in the normal skins are covering the epidermis, generating our skin tone. You see that melanoma is retained this ability of producing those melanosomes, and it's a lot of energy. So the big question is why? Why would keep generating this differentiation feature of the origin cell in melanoma? And this is something that was raising a lot of questions during my postdoc, and when I started the lab, we started investigating it. So we were showing, and the work that I'm showing you now with the great collaborators, is that melanosomes are not anymore spread in the melanoma, are not anymore just spread in the epidermis. Now they are losing their orientation and start being spread down also to the dermis. And we found interactions with CAF in collaboration with Neta Erez. So we found that those melanosomes are being uptaken by the dermal fibroblast, uptaken and secreted in distance metastasis, and this is a great collaboration with Ron Shamir and Dvir Netaneli. And that was a great paper because Dvir was approaching me and say, I see this weird signature of pigment in those, it's a computational lab, in patients, can you explain it? And we figure out that it goes along with what we were showing in the lab, that those melanosomes are basically shaping the microenvironment and supporting the tumor progression. And then we also published recently with Shoshi Greenberger in Sheba that those melanosomes are also been uptaken by the dermal lympha, so lymphogenesis, generating the lymphogenesis of the dermis. So all of these three works are talking about the next metastatic niche of the tumor in the microenvironment and supporting them. There is also another group showing that those melanosomes are also taking out chemotherapy from the tumor cells, supporting, again, tumor progression. So these were the works that we were showing, and again, mostly metastatic niche, so how melanosomes are supporting. They are not like exosomes. Exosomes are being spread systemically, but those melanosomes are being spread in the close proximity of melanoma and shaping the environment of melanoma. And we raised the question of, so what else those melanosomes can do in the tumor microenvironment? Now, I'm going back to this basic picture, pathology, and it's about, sitting with pathologies like Dov Ershkovitz and Eva and Hanan Vaknin from Wolfson, and I love sitting with pathologists because all the secrets are there. So when you look at this image again, you can appreciate how just below melanoma there is concentrations of lymphocytes and macrophages. So there is an immune recognition of the tumor. And based on this immune recognition of melanoma, which is highly, with high mutation load, etc., so there are many interesting interaction between melanoma and immune system. And then I decided, okay, so let's start studying some immune features and immune interaction with the immune cells. And thanks to Asaf and Yaron, who led me into the immunology world, we started investigating it. This is the work we published last year, and it goes along a little bit with what you were saying. So what we were showing here is that macrophage in the environment of the tumor, uptaking melanosomes from various types of cells, and this dictated a different type of macrophages. So macrophages are similar to fibroblasts in what we learned from you, that you have a variety of macrophages in the tumor microenvironment, some pro-inflammatory, some anti, some remodeler, some cleaner, so all these very basic terms. But what we were showing here is the triggers. So we were showing that macrophages, you see here in the cover we designed it, so macrophages are uptaking either melanosomes that are breaking from the epidermis. So you see how there are cells here that have melanosomes, and when they are break, those melanosomes are uptaken by macrophages, or dermal cells. So also fibroblasts who has melanosomes inside can be break and secrete their melanosomes. So basically, macrophages here in the center of the tumor receiving melanosomes either that they were break from epidermal cells, keratinocytes, or from dermal cells. And then they are eating different dishes. And because they are eating different dishes, and we were showing how they are different, those melanosomes, this is how macrophages are polarized to different state inside the tumor microenvironment. So we were bringing up an idea of how you polarize a very plastic cells inside the tumor microenvironment into different features. So that was in this paper. But what else? So what else is there with melanosomes in the tumor microenvironment? Of course, I'm going to go through CD8 cells, okay? The recognition of the tumor by the immune system is the basic of generating a lot of immune therapy. And it revolutionized, as we all know, cancer and cancer treatment in the past 12 years, right? More than a decade. But still, 50% of people will not respond to immune therapy. There is something that the tumor can overcome, even if the tumor is recognized by CD8. So even if there are lymphocytes inside the tumor, and tumor-infiltrating lymphocytes can be found in a tumor, still this tumor, although being hot or inflamed, will not respond to immune therapy. So there is something that blocks the ability of the T cell to kill the tumor cells. And that was the question that we were raising here. So what can it be? Now, when I start reading papers around it and looking at this field of like cancer escape mechanism, one of the papers that was dealing with RNA-seq of patients that respond and non-responders to immune therapy, I came across this paper by Riaz, And there was hidden in one figure in the paper, nothing was highlighted there, but there were 3 lines about melanogenesis, so the production of melanosome. And in this paper, they were actually mentioning that there is upregulation, okay, of melanosome signature in these non-responders of melanoma to immune therapy. And I said, okay, so maybe melanosomes are taking part in this non-responding into immune therapy in the tumor microenvironment. We then searched the literature, and I'm going to go very, very briefly. But now when you have a different vision in your eye, so you can read old papers with a different Google, and then you can see those things that are just there. So people were reporting that melanosomes is a bad prognosis. It is in non-responders, you can find it. It is going up and down as melanoma is progressing, as shown here. If you block melanogenesis or melanosome production, a T cell will kill better melanoma. And this is proteomics that we did on melanosomes and exosomes. So we gather all of this information, and we came up with the hypothesis that, okay, maybe melanosomes play a role in the immunity of melanoma in terms of recognition and the ability of CD8 to kill melanoma cells. Here I approached Mitch. He has an amazing biobank in Zurich, and he gave us a tissue array of responders and non-responders of melanoma. And we did a very simple staining here. We stained for pigment, just pigment, not even an immune staining of melanosomes. And what was striking was that when you look at those pathologies, and it's 100 different patients there, I'm showing you here several, and you look at their lymphocytes, we found this ring of pigment around the lymphocytes of non-responders. So people that do not respond to immune therapy, their lymphocytes were enriched with this ring around them, just in the membrane of the cells, while it was not that much of a peer in the responders to immune therapy. And then we said, okay, so maybe we have something here. We came now to the pathology of melanoma, and now we stain with specific markers for melanosomes. And you can appreciate here, this is a CD8 marker, and this is melanosome marker, and it's like a ring. So melanosomes are coming, binding to the membrane of the CD8. Not all of the CD8, which was also encouraging, not all of the CD8 have those melanosomes. So then we start feeling that there is something specific. So melanosomes are targeting the membrane of the T cell, but not all the population of T cell. Then we approached the Bar-Ilan Institute in Sheba, and here in collaboration with Orit and Michal Besser back then it was, and now it's Ronnie and Yochai that we are working with. So we approached them, and then we received samples. So they have TIL, tumor-infiltrated lymphocytes, and melanoma of the same patient. And what we did, we were isolating melanosomes, and then we were seeing this interaction between the TIL and the melanoma in the presence of melanosomes, and to see whether those melanosomes will interfere with the ability of T cell to kill melanoma. And that's what you can see here. You see significantly less killing of melanoma, less secretion of interferon gamma, which is indicative of T cell working less. And when melanosomes were around, they were working less. Then we moved to an in vivo model. So we injected melanoma, and we injected intratumor a bleaching material. So in the East, there are many investigations for bleaching, and we were using one of the drugs for bleaching of the skin. And this drug, we validated it. Indeed, it is blocking melanosome secretion, melanosome production. There is less pigmentation. So we injected it intratumor, and you can see here significantly the tumor is shrinking when there is less melanosome being secreted out. We have the control to show that the tumor itself is less pigmented. Okay? There is less secretion and production of melanosomes, and there is an increase of CD8 infiltration into the tumor. Here is more controls just to show that this drug by itself is not affecting the proliferative of melanoma cells or the T cells. So there was something specific about the ability of melanoma to produce and release those melanosomes, and their ability to enhance the ability of CD8 to react to the tumor, to proliferate as a response for the reaction and recognition and killing of melanoma. So that was in vivo. And then we said, okay, so let's go deep into the mechanism of how those melanosomes are doing it. We did mass spectrometry, proteomics of melanosomes and exosomes from the same melanoma. We did it on several melanoma lines that we have in the lab, mouse and human. And we found that melanosomes carry a very strong signature of immune-related proteins. You can see also the pigment-related proteins. And what was also there is that melanosomes carry antigen presentation ability. So the proteins that are involved in antigen presentation. And what does it mean to the non-immunologists in the audience? So when tumor and T cells are coming into interaction, okay, there is this very specific interaction, which is the TCR, the T cell receptor, is interacting with MHC, that is presenting antigen to the T cell. And this is the specific recognition of T cell to the tumor. And we found this complex presented in the proteomics of melanosomes. So basically melanoma is secreting vesicles or secreting HLA out, secreting MHC out of the tumor to the environment. Then we were trying to investigate whether the interaction between melanosomes, okay, and the T cell is TCR dependent, this MHC TCR dependency. And it was indeed. You can see here how melanosomes are interacting with the T cell. You can see here we compare the interaction of T cell with melanosomes compared to exosomes. And again, melanosomes are binding to the membrane of the cells, while exosomes are being uptaken by the T cell. So there was something, again, specific and indicative of membrane-to-membrane interaction between the cells via the TCR, while exosomes are being uptaken, as exosomes know to do, and this is with lymphocytes, with the CD8. So the interaction was TCR specific. Here we were also showing using different type of mouse models. Here's the GP100 that we received from Michal Lotem. And we were using them, and again we were showing that the interaction was the ability of melanoma to react to those CD8, or the CD8 ability to kill melanoma, was dependent on melanosomes that can be recognized by the TCR MHC or HLA. Here we were showing with an antibody that those melanosomes indeed carry it, and this is upregulated after interferon gamma. And when we eliminated the MHC from those melanosomes, the interaction between T cell and melanosomes fell down significantly. So the interaction was specific to the MHC and the ability of the T cells to be 3 minutes. Yeah, okay. So I told you that the HLA MHC is presenting antigen. So we then approached Yardena Samuels. She's doing HLA peptidomics, and we gave her those melanosomes. It's the first time that vesicles, extracellular vesicles, this HLA peptidomics is being made, and she did it. And what was striking was that when you give her melanoma cells and their HLA or MHC, or melanosomes from the same cell source, different antigens are there in the sequence. You can see it here that there is a higher interaction. So the affinity is higher in the melanosome antigens. And also here with Pierre Close, when he analyzed the sequence, he saw that the peptides that are presented by melanosomes are more immunogenic. They conceive more missense mutations, which means that they are more attractive to the T cell. They have more potential to attract the T cell. Here is some other direction that we are taking now. This is together with Dvir Gur from Weizmann. We are looking at the membrane of those melanosomes to try to figure out how this MHC complex is being uploaded into the melanosomes. And here with Michal Sharon, also at the Weizmann, what we found, and that was motivated by, after a talk by Neta Regev, we found that also those melanosomes carry proteasome, and not any proteasome, they carry immune proteasome, an active one. So maybe, okay, those melanosomes are not just presenting their antigens, but they also carry independent form of proteasome by means that they are digesting the peptides and have like their own system. one more slide about the mechanism. We were trying to see how the melanosomes are affecting the T cell. Here we did RNA sect of T cells that are receiving melanosomes and interacting with them, and we saw a very confused signature, which means that the T cells don't know what they interact with. So they are being interacted via their TCR, and then it's a paper about the signal 1, signal 2 by Dino that we were reading carefully. And so there is a confusion between signal 1 and signal 2 of the T cell, and indeed the interaction with melanosomes is leading to death of the CD8, while interaction with melanoma is not. So those CD8 are very happy when they are with melanoma because they recognize and kill it, while if melanosomes are around, they are getting confused and eventually they will be apoptotic. So that's what I was showing you today. Melanoma is spreading those melanosomes to the area, and then those melanosomes are interacting with the subsets of T cells while other T cells are recognizing melanoma. And because we are losing interaction with the T cell, then there is we can explain the non-responsive to immune therapy. There is so many people to say thank you, and many, many collaborators. I mentioned few, and if I forgot anyone, forgive me. But we couldn't have done it without those great collaboration. Thank you all.
Thank you, Kermit. Fascinating talk. Any questions from, okay. Lots of hands raised. Michael, and then.
Kermit, very interesting. UV light is known to be a strong inducer of inflammation, and how you would, and obviously, you know, skin cancer is not just melanoma, but also basal cell carcinoma and squamous cell carcinoma. Basal cell carcinoma is relatively easy to cure. So do you think it's somehow regulated? So I try to understand if this is, those melanocytes are melanoma-specific phenomenon, or under UV, melanocyte will secrete those
melanosomes?
vesicles? Yes.
It's a very, very good question. When we compare the proteomics of normal melanosomes from normal melanocytes and from melanoma, we saw differences. There is a different microRNA inside, different proteins inside. Okay? So it might be different, but it might be true in vitiligo. So non-related to UV, maybe there are specific melanosomes that will do the opposite. Then if they have the ability to interact with the CD8, maybe they will educate them and will react to vitiligo. So this is something we are thinking about. We haven't done anything with it. With the UV, maybe there is a massive secretion of those melanosomes. There is an inflammation, but there is also a very great repression of the immune system in the skin. This is what you do phototherapy to autoimmune. So it's like the strongest will be the phenotype that will dictate. So it's mostly repression with those melanosomes that are being more secreted after UV. Then I think this is the combination. So maybe it is answering a question for something specific to melanoma. But the ability to secrete large vesicles with all of these cues, I think it's general.
Dino? Beautiful talk. I come in here. Do you have a way to capture those T cells that are interacting with the melanosomes as opposed to the ones with the tumor cells and see the phenotype?
I hope you are not my reviewer. Okay? So it's a wonderful question. We will have to deal with it. So we have specific antigens. We have specific, because we have the signature of what melanosomes are presenting. Now we are repeating it with many more tumor cells to see if we can catch a very specific something that we can dig. And this is what we're trying. We will try to pull out those T cells. What we are thinking to do with Sheba, for now, just therapeutic-wise, is to have plates covered with melanosomes and try to clean TILs before transplant them into the patient, try to kill the TILs that are binding to melanosome, and then you will have a pure population. So, but that wouldn't be able to what you want. So we are trying to find a specific one, so we will be able to pull it out. Yochai, what did you want to say?
What about CD4 T cells?
What about CD4 T cells?
Wonderful question. I don't know. So in the tiers that we are receiving from you guys, it's a
Just
mix.
repeat the question again.
So what about the CD4, if there is an
40.
interaction?
CD40.
CD4? Yes. Okay, so I don't know. It's a great question. From the tier that we are receiving from you guys, we have a mix of CD4 and CD8. Orit did some facts. Sharona, she did some facts on those CD4. I still haven't touched the data. So the data is there. There is some effect, but it will be hard for me to say who is, like, what is the sequence of events. It's a great question. We are very much interested in that, but not in this yet.
I'll leave the rest of the questions to the break, being mindful of time. Thank you, Karmit.
Thank you.
Last but not least for this session, I would like to invite Professor Shai Shenor from Technion, who will speak about charting immune landscape dynamics. Shai?
Thank you for the invitation to speak on this important, I guess, Cancer Day. For disclosure, I'm also the chief scientist of Cytoreason, which is a company that applies much of the tools we developed in the lab in the kind of real world of drug development. And what I want to talk about today, and I kind of changed a bit the title of the talk, is something that's been a growing interest of the lab and relates to something that we take for granted and ultimately to the point where we actually ignore it. And this is how to think about dynamics. Now, I tell my students often that I think as academics, we have the luxury of reading and doing science, not just in our domain, but also good scientists would read breadth and get inspiration into their own field. And the example I'll tell them is you should be reading papers about dinosaurs, right? And the reason I say this is because this topic of thinking about dynamics was inspired by this particular kind of view of what of the Oldman Formation in Alberta. This is in Canada. This is a 40-meter deposit of dinosaurs. And why would this be interesting? Well, back in 1975, there were discoveries there of what was thought to be 3 genres and 12 species of dinosaurs. And back in 1975, a paper came out saying, look, guys, if we look at these skulls of dinosaurs and we model that dinosaurs are also grow with time, then we only have 3 species of dinosaurs here. The rest are just baby dinosaurs, right? And it was a single paper that then extrapolated into this entire field that we actually have way less dinosaur species than we imagined because we're not modeling the fact that dinosaurs change over time. And with that idea, I would argue that the same problem exists in medicine. So time is an essential component of how we should be thinking about disease, but it's really difficult to do. We struggle with it, and ultimately, often, I think we give up on it. We don't put it well, in a good way into the equation. At different levels, you can think about short time spans like cellular differentiation. You can think about longer time frameworks as treatment response. You can think about really long time towards as disease progression and so forth. And in my own lab, as people may know, we work on immune aging, which is basically a 90-year-old pathway. And so the reason is kind of obvious. To actually take time into equation, you need to do really long experiments. You usually don't know what's the right time points in which you profile technologies, kind of most recent technologies around omics, really, to get the data that you want, you destroy the sample so you can't follow samples over time. And then just all the intermediate stuff gets lost, right? And so there's been kind of solutions around this which are trying to basically recapture something around time, and that is using some kind of computational methods, and people may have heard the term pseudotime, right? Which is our attempt to understand what's happening when we don't have a good measurement of time. The problem is that if you really look at how science moves forward, it moves forward when we have quantitative frameworks and abilities to basically describe a language of comparison. And so if I want to compare two things, always there's a control and so forth, I need somehow to be able to describe this in a very good sense. And so the question that was been posed in my lab, and we've been working in a lot of different directions on it, is how to make biological processes, things that change over time, quantifiable, okay? It can be aging, it could be disease progression, cell differentiation, and so forth. And I'll tell you where we got inspiration from, and this is sequence comparison. Both from a algorithmic perspective, because we've been kind of taking those old algorithms and revisiting them with respect to time, but also from a language perspective and the revolution that they offered when we developed the tools to compare quantitatively two sequences. Suddenly molecular biology moved forward quickly because I could do very complex comparisons and infer them. And so the tools here are around a lot what's called dynamic programming and so forth, and this is just showing you one of the famous algorithms called Needleman and Wunsch, where you're looking for a pathway and its comparison to compare two pathways of two sequences. And the same thing could be done in temporal processes, and so I mentioned people build trajectories all the time with single cell data, but I'm sure people here are well aware of this. There's now over 200 different tools to build trajectories, but actually very few on how you compare trajectories. So a trajectory is a process that changes over time. How can I compare my trajectory to yours, right? My biological process to yours. And so we started this journey a while ago, back in 2018, and have been publishing around cancer, which is the focus today. Started in 2022. This is work led by Ayelet Alpert, who was an MD PhD student in the lab. And to kind of show you here, imagine two conditions, A and B. I have single cell data. The blue cells I can order, and I can look at a gene here and how the expression kind of changes over time on the top plot, and I can do the same on the bottom one for the other condition, and you can see that the expression of the gene looks differently, but there's some similarities, and what I want to do is actually compare the two, and so what we've developed is a framework that uses dynamic programming to basically compare one pseudotype to another and find where the similarities are, and can we stretch the pseudotype and so forth to find the best alignment. If you like, you can do this with global alignment, local alignment, so the entire trajectory or just areas that are similar, and it's a quantitative framework to really get higher resolution comparison between two single cell trajectories. And in cancer, we call this the tumor axis, so you can think about these conditions in where I have two samples, and I want to compare the trajectories of development, and the problem is that, of course, that in cancer there's a lot of different mutations, and ultimately it's actually very hard to compare the two processes. And so we built a framework that allows me to compare a cancer sample to either another cancer sample or to a reference healthy sample, where I get different weights based on the conservation of the markers, of the conservation of the genes that are being used in the process. So ones that are highly conserved actually get a high weight, and they therefore have a high impact on the alignment. So to kind of show you the implications here, this is work, kind of one of the many validations we did in this work, looking at AML of a cohort of patients we collaborated with Ishaya Ofran on. Well, I guess we'll be speaking here later today. No, not speaking here later today. Oh, okay, sorry to hear. Basically what we took is we took 9 healthy individuals for which we got AML samples, and we got bone marrow samples, and then another 21 patients with multiple time points of diagnosis, day 14 and relapse, and we profiled their bone marrow by cytoff, and just to kind of show you here on the right-hand side, you can see that you basically get this very kind of, I'm showing this a low resolution trajectory of myeloid development in the bone marrow. Right? Now, I can also now take this trajectory, look at the healthy samples to say this is what myeloid development looks like in healthy samples. Now, what are the different markers? How do they behave in the healthy, and how much weight should they have in the AML samples. And so we do this for each one of the different markers. And we can see how you can see the generally progression where I can assemble a trajectory of healthy samples of the myeloid development in the healthy samples. Now I take each one of the different AML patients, right? And each of them actually has a problem where they have some form of AML. We've characterized it genetically. We've done all the kind of standard flow resolution, but we have the standard flow markers, but we have a very high resolution 40 marker panel for cytoff on them. And now we can take each one at a single cell level, align the trajectory, and describe a patient not based on their genetics or their flow markers, but actually describing how their sample looks like on this healthy trajectory, right? So if you look at the healthy individuals, they kind of shown here in the black line, you can see this is how myeloid development looks like in the healthy, and you can see the different AML patients the healthy individuals and so forth, and you can see the way that we can now take this trajectory and for every AML patient now describe how they shift along this, right? And the way we do this is actually look at what's called Earth mover's distance or optimal transport of how much would it take me to move from the healthy sample characterization along the trajectory from the AML to the healthy. And this is the heat map showing you the trajectory of healthy, so the two map pseudotime we're aligning to from 0 to 1. And this is just shown as a color plot. Every row here is one patient's AML sample, okay? And you can see that they look markedly different from the healthy, right? And they look markedly different from one another. So we now have a new way of stratifying myeloid kind of abnormalities in AML based on how the differentiation looks like at a single cell resolution, okay? And we call this the stemness index, and you can see as an example here that a patient's stemness index would actually shift over time with the treatment. And in fact, this is different between what you would see with kind of a diagnosis in day 14 or day 14 and relapse and so forth. So is this important to have as another yet characterization of AML? Our answer, and this is obviously a small sample size, is yes, because now we can take individuals, look at their stemness index, and describe their ability for basically their survival based not just on the clinical factors and standard genetic markers that are used in AML, but also when putting their stemness index into play in the model. You can kind of see it here. The p-value without the two-map algorithm is significantly kind of smaller than it is when we're actually taking this into account and are able to stratify the patients at low and high risk just based on their trajectory of development. I'll take this to the next step, which is rather than thinking about single cell, the much more relevant clinical case where we're looking at multiple time points. In say bulk expression, this is what's most commonly collected still in clinical trials and in investigator-initiated clinical trials. And the work we did here, led by Amit Frischberg and Netta Millman, Netta was a postdoc in the lab and formerly here at Tel Aviv University, was around how do we actually try and build large-scale temporal processes from small time points of intervals of samples. So you can see here the problem. I have a bunch of patients collected. I may have some time points for them, but again, nobody follows a patient for a long time. Nobody's collecting the right time points. Who knows when the right time points are? So I have multiple disease stages, and I have, and really what I'm trying to collect to get an understanding is how does this disease change over time, right? And the answer here, as I was saying before, is just like we were leveraging standard sequence alignment for the single cell. What we did here is build a multiple sequence alignment, okay? So leveraging on the same ideas that come here. You can think about every patient coming into this with having some, time points. Our minimum is 3 time points collected per patient, but no one is being tracked for the entirety of the disease or has high resolution data. And what we do is we use multiple trajectory alignment. We call this Timex. Oh, 3 minutes. Oh, okay. to then build a consensus trajectory of how the disease changes over time. We're able to say how every patient differs from that trajectory, and we can take a patient and project them to give you a pseudotime on it. So just to show you here, if I'm looking, we took a small data set of bladder cancer, 18 patients, 84 samples, quite rare, where you can basically, patients were followed up to 15 years. And we run this process of Timex where we build a consensus trajectory of how bladder cancer looks like. This is RNA-seq data, and then we validate this on a variety of other publicly available data sets. So we can just show you here, you're looking at the time from primary tumor years on the x-axis. Here. And this is the disease pseudotime. Notice the huge difference between how much time chronologically has passed and what molecularly is happening in the RNA-seq sample, okay? And the variation between clinical staging, so stage 1, 2, 3 of the disease, and the variation in what's happening molecularly, right? That's the x-axis. The y-axis here, the disease pseudotime. And we can now take those patients and on the time x-axis, position each one to look what they look like. So here's the disease pseudotime, and we estimate the purity of the sample. So what's the percent of the tumor in the sample? And you can see a huge shift over the time x trajectory with having this inflection point in the middle where tumor purity drops, and we've deconvolved this to show there's a strong infiltration of immune cells at this time. And if you're just taking the standard primary data of the time from since primary tumor, you just basically get nothing out. Is this useful? Well, I can now take the TCGA, bladder cancer, about 430 samples, and use this kind of point of position every patient in TCGA. They only have single time point, but I used 18 individuals to build an entire trajectory. And now I can position, basically project patients on them, and pre and post this inflection point, the stromal point of infiltration. You can see this actually helps statistically in being able to predict survival. Even when I just take subsets of patients like that have been characterized with basal squamous carcinoma, I can identify significant differences in survival. And the same goes if I now take genetic phenotypes of molecularly described genetic phenotypes of bladder cancer, and try and actually look to see, can I tell any more additional information based on the molecular positioning on the time x trajectory. So just showing you here, patients could be primary, this could be a primary recurrent tumor. The variation in the disease pseudotime, the time x-axis can be huge within those. And it has basically very little to do with the molecular phenotypes, which are not really used clinically because they don't provide much value. But look at URA, huge amount of variation in the molecular phenotype, and this correlates their position within URA on the trajectory correlates with survival in the TCGA samples. And this can also be used mechanistically for characterizing what is happening in the tumor. So I can take URA samples, characterize which one have an early pseudotime, which one have a late pseudotime. Those from a standard bladder cancer known molecular markers, there's no difference between them. You can kind of see it here. These are the kind of hallmark genes of in bladder cancer. But now characterizing, are there differences along the temporal variation of the time x-axis? Can I find basically new functionality that differs between late and early pseudotime? What we've been able to show is that there's a module of downregulated pseudotime genes that happens with the trajectory. All of those pseudotime genes are basically the antisense to cancer proliferation genes. And on the upregulation with this are very known kind of cancer hallmarks, but also never noted before, a mitotic kinetochore spindle assembly genes, all of which are correlated with kind of late versus early discovery of the late versus early bladder cancer positioning. So basically, by characterizing temporal variation, we can discover new biology that's basically missed if you just take your kind of samples one place at a time, and we can describe at a very high resolution what's happening in every patient. I'll summarize here and say this, I kind of said it in the beginning. Dynamical processes are basically that information is underutilized in the way we do our science. There's tools to do this now that we've been developing, and I think I kind of highlighted a few of them. They're key to this, and happy to take questions. Thank you.
Thank you, Shai. Beautiful talk. Question?
Thanks, Shai. Beautiful. So maybe I missed this, but how much of what you see is dependent on the cellular composition? So you mentioned like the immune infiltration point where you see things changing, and how much of it is the actual genetics? And then as a follow-up question, how different is it between tumors with like high mutational burden, tumor types, versus other tumor types?
It's a wonderful question. So for lack of time, I didn't show the entire cell composition, but we model the cell composition, as you can imagine. We like that type of stuff. Within the Timex trajectory, you can see shifts within this. The infiltration point I showed you is a huge kind of shift in migration. But much of the analysis, what I actually showed you with the late and early molecular phenotype, that's all taken pre-infiltration. So we just to kind of make sure we're not kind of being biased by cellular information. We're looking at the differences with it where there are no significant changes in immune infiltration, suggesting that we're picking up biology that isn't due to cell composition. We've also done, we've developed techniques where you can model the cell composition in RNA-seq. You have that information, you remove that bias from the data, and you're looking at regulatory programs that go beyond this, and you see significant differences there as well. And with respect to high versus low, we're now running this across all TCGA, so I'll let you know.
Two more questions. Vered?
Beautiful talk, amazing. I was wondering, have you tried to validate this on a prospective cohort rather than using like the one time point?
Yeah, so we've done, so the validation here was really to show that we've done this now in publicly available data sets. We've done this also on, not in this lab context, on clinical trials from pharma, which I wasn't seeing before, and could repeatedly being able to validate and show better stratification.
Carmen?
Great talk. I guess it's an obvious question, but what is the age of the person, or the age of their cells, their cancer cells, is affecting the trajectory? So if it's an, so taking the time of regular aging into the account, so that's, and then the other question is, no, I'm sure, this question.
It's a good
okay,
question.
Let's start with this one.
I didn't say it, my bad. All of the analysis I showed you, we correct, we adjust for demographics of age, tumor staging, gender, and so I feel it's not due to this. We do have a paper that's just coming out now with Mark Davis in PNAS where we're showing across the immune aging trajectory that if you get to around the age of 80, you're going to start picking up immune signals that are predictive of cancer, but that's not in relation to the Timex.
And the trajectory is always one way?
No.
Like you don't see?
No. We have, we're basically, the way we build this allows us to we're biasing towards kind of moving progress, but no, we've published papers where we see patients in COVID say that are moving back on the trajectory, and so there's a lot of characterization. We're doing this also now in sepsis, patients who move to recovery versus not. So you can tell, it just adds a whole new dimension.
Really innovative. Thank you. I'm sure there are plenty of questions. We'll have the break now. Just before we go to the break, I would like to thank all the speakers of the first session. We're looking forward to the second session. Before that, we have a picture for all the speakers. So please join me here on the podium, and we'll go for a break. We'll come back in 15 minutes. I'm really excited about the next session with this great lineup of speakers in continuation of the previous one. The next speaker is Professor Yossi Yarden from the Weizmann Institute of Science, who will speak about acquired patient resistance in anti-cancer therapies. Thank you, Yossi.
Thank you. I'm happy to be here and meet so many young people interested in oncology. I know it is Cancer Day, but I want to go back to HIV/AIDS because this is a successful way of mankind to face epidemic and a medical issue. And there are some lessons. Even though it is an epidemic, in a way, it is not anymore a problem because, even though there are more than 1 million people who were infected, it is not a reason for death. And even though no patient or very few patients can claim that they were cured, still, and there is no single drugs that can take care of the disease, still a combination of drug is sufficient, and the disease is chronically existing. And I think that this might be a lesson for us when we come to dealing with cancer and long-term treatment. So several types of cancer, including blood cancer, are now being treated with chronic strategies that are similar to what is the situation with diabetes or other chronic diseases. So cancer incidence is indeed going down in male and female since the mid-1990s. And as you can see here, started around 1990, 1995, and this is primarily due to early detection and maybe also prevention. As you can see here, therapy is playing a role, maybe not a major role, and you can see here the history of treatment of cancer going back to the mid-70th century, surgery, radiotherapy, chemotherapy, all of them remain with us, except they are being refined and more effective and less toxic. And today I'm going to speak about oncogene or mutation-targeted therapy, primarily resistance to the new drugs. And of course, you heard before, the era of immune checkpoint blockers like PD-1, PD-L1, CTLA-4. So in the oncogene-targeted therapy, there are primarily two classes. These are the tyrosine kinase inhibitors, which are small molecules, synthetic, and they induce rapid resistance, as I will be showing you later. And the monoclonal antibodies on the other side that are recombinant, big molecules, and resistance also occurs, but in a little longer time. So some issues that emerge when oncogene-targeted, mutation-targeted therapies became second to chemotherapy. So the kinase inhibitors are usually used alone, unlike the monoclonal antibodies that are usually combined with either chemotherapy and rarely, but still, with radiotherapy. Intratumoral genetic heterogeneity complicates cancer treatment because we relate to the tumor as a collage of several tumors that are different in terms of the genetic makeup. And some of the most frequently mutated genes, P53 in the first place, appear to be non-druggable, at least with the chemistry that is available today. And today's lecture will relate to secondary emergent resistance that limit drug application. I'm sorry. Okay. And I would like to take you to the mathematical approach to understanding and improving cancer therapy, primarily because we relate to biological networks and the generation and the area of mathematics that deal with network and the ability to inhibit them in silico, so-called graph theory. So while network expand, rich nodes become richer to the extent that we see some addiction to rich nodes. Networks are trained to resist common perturbation, but they show fragility to uncommon perturbation. And I will give you several examples. Robust networks are Hub addicted, such that uncommon interceptors, drugs, targeting major hubs may collapse the networks. And there is a principle which is interesting, which is hub centrality breeds lethality. I want to show you an example. This is a network with several hubs shown in red and several connectors here, if you want proteins or metabolites. And now we would like to target the network because it is oncogenic. So if we now go to peripheral nodes, the network is still robust. What we need to do is to find a central hub that when we target it, the network collapses. So the principles are find a hub that connects to many peripheral nodes, like EGFR that I will show later. Also find an uncommon perturbation that was not tried in the evolution of the network, and I will relate to antibodies. Also, come up with simultaneous inhibition of compensatory pathways, because when we try to target hubs, several compensatory hubs come up. So we must go to combination therapy. I would like to show you the plasticity and the upregulation of hubs after we treat one node or maybe a central node. So in this case, we are dealing with the use of antibodies to EGFR in order to inhibit a mutation that activates EGFR. You see, phosphorylated EGFR. So the antibody is working well, downregulating the receptor, and the receptor disappears. Now the phosphorylation is decreasing initially, but then you see upregulation of phosphorylation. And this is due to the compensatory hubs that transphosphorylate EGF receptor. These hubs are in fact brothers of EGF receptor, HER2 and HER3, that are epigenetically upregulated and transphosphorylate EGF receptor. Now, if we go downstream to the MAP kinase pathway, which is a major one, you see that initially the antibody is active, but phosphorylated ERK is strongly activated, much more than the initial level. So the network is fighting back. And what we can do, as I told you, we can inhibit the other hubs, the compensatory hubs. When we do this by inhibiting HER2 or inhibiting HER3, you can see that this overshoot is lower. When we inhibit all three of them, the overshoot disappears. And this is due, we think, to the disappearance, to endocytosis of the receptors, EGFR in green, HER2 in red, or also HER3. And in the end, the cancer cell remains stripped from all this hub, for all these receptors that are oncogenic, collaborating with EGF receptor. So going back to lung cancer, as you know, lung cancer is more prevalent and more lethal than the next 2 or 3 other types of cancer in the list that Ronit showed us this morning. There are mutations in KRAS, primarily in smokers, but there are also mutations in EGF receptor. In Israel, it is about 12%. In Asia, it might get up to 35–40%. And there are good inhibitors, tyrosine kinase inhibitors, which are ATP mimetic, penetrate into the ATP binding site and stop catalysis and phosphorylation, let's say MAP kinase. So these drugs were first approved in 2003, and the last one, the latest one, was approved just a few months ago. And the reason why we have so many drugs, and the reason is that when we apply the inhibitors like gefitinib, erlotinib, and the latest one, osimertinib, resistance in terms of new mutation appear. And the mutation nullifies the drug because the drug cannot see EGF receptor, the target. And this occurs once and again, in a matter of a year average per patient. And this is the latest resistance, C797S, that preempt osimertinib, which is a covalent inhibitor, very sophisticated, very effective, except patient become resistant in a year or maybe a year and a half. The appearance of resistance and the mutation, as you can see here, is preceded by upregulation of AXL, AXL, and several other receptors collectively conferring a phenotype similar to EMT, some of you may know. So we thought that if we inhibit AXL, it might help to prevent the resistance appearance. We collaborated with a group in San Francisco that showed that single-cell RNA sequencing of patients who were treated with osimertinib, the latest drug, in those single RNA sequencing, you see upregulation of AXL in the residual disease and further upregulation in progression disease, resistance tumors. AXL is a cell surface receptor. It binds to GAS6, and GAS6 binds to, which is here, GAS6 binds to phosphatidylserine that is otherwise protected from outside. When cells undergo apoptosis, phosphatidylserine is exposed, bind to GAS6, and activate AXL. So we thought that maybe AXL acts as a sensor of cell death, of apoptosis. This is a collaboration with Gadi Getz, who looked at the persistent cells, thought that are not yet mutated, but they undergo upregulation of AXL, and he asked what is the upregulation, AXL, and what else? GAS6 was the first one over here. So we were convinced that maybe AXL and GAS6 play a role, and in the next step, we knocked out AXL, and then used in vivo model in which cells, these are tumor cells, cancer cells, are inoculated subcutaneously and either untreated, this is a very aggressive tumor, or treated with the first generation inhibitor in green or the latest inhibitor in red. And you can see that there is resistance, there is response, sensitivity, and then upregulation due to relapses, to the appearance of the mutation. When we used AXL knockout cells, same cells, except no AXL, you see that there is no relapses, either after treating with the latest kinase inhibitor or the previous one. So AXL is probably mediating the relapses. We reasoned that the extensive apoptosis that precede the emergence of the new mutation and relapses, this might have to do with the relapse, of course, and the connection between extensive cell death and relapse resistance is known since the antibiotics were studied and were in the focus of research. And work done by scientists working in the 1970s and 1980s, they came up with the notion of an SOS-like response that occurs in bacteria treated with very effective antibiotic. This machinery, the SOS, bacterial SOS, activates several genes by means of promoter activation that induce DNA repair downregulation, and on the other hand, mutagenesis that is upregulated. And the way it does so is primarily by going to the replication fork, the DNA replication fork, and inducing upregulation of sloppy, mutation-prone DNA polymerases. I want to show it to you. So this is the DNA replication fork, and what we found that there are two types of mutagens that are activated when we apply the very effective kinase inhibitors. One of them has to do with the availability of purin versus pyrimidine. There is a metabolic switch that overexpress or make purin more available, and this causes mutation. Also, there is recruitment of the mutagenic polymerases coordinated by a protein called RAD18, which is an E3 ubiquitin ligase. In reality, it looks like this. DNA replication occurs normally as there is no genotoxic stress. So if there are mutations, they are corrected, and there is pCNA here that you can see as a ring-like molecule. Upon genotoxic stress, there is recruitment to the DNA replication fork of the mutagenic polymerases. They introduce mutations in order to provide a way out of the signal to death, to cell death. So they blindly introduce mutations, like in here, in red, and in the next step, probably those mutations will be corrected or not. I want to show you this occurring in our cancer cells. This is by PCR. And in her, this is in vivo, in which every lane here corresponds to one mouse. And here we treat with the kinase inhibitors with AXL overexpression. And as you can see, after we apply the kinase inhibitor, there is downregulation of many DNA repair enzymes, because it doesn't make sense to introduce new mutation and repair them, right? So there is downregulation of the DNA machinery, of the DNA repair machinery, like MMR, like BRCA, and upregulation of the mutagenic polymerases. You can see them here, many of them. In parallel, there is downregulation of the proficient DNA polymerases. And in animals, it looks similar. In here, you can see the upregulation of the mutagenic polymerases in the mice. And there is the downregulation of the proficient DNA polymerases and downregulation of DNA repair as well. So we made an antibody to AXL. We cannot make a knockout, right? Of course. So we made an antibody to AXL, hoping that it will inhibit AXL and the mutagenic process. And as you can see here in green, this is the same in vivo system, treating with a kinase inhibitor over time, and tumors first shrink and then relapse. So the antibody, the new antibody, was not really effective when treating alone. Also, when in combination with anti-EGF receptor antibody or with a kinase inhibitor, it became active only when we treated triple combination. In her, you can see that the combination of the new antibody, anti-AXL, anti-EGF receptor, and the kinase inhibitor, there was no relapse, like the knockout I showed you before. At this point of time, we were looking for feedback from industry, for pharma companies. And we went to Pfizer, we went to Merck, to Roche. What would be needed in order to make it attractive in the eyes of the pharma companies? And the feedback was double. One feedback said, why do you use two antibodies, two kinase inhibitor, one to AXL, one to EGF receptor? Why don't you combine two kinase inhibitors? It is easier for us to give a kinase inhibitor. It is taken as a pill in the morning. Taking a monoclonal antibody is cumbersome, right? You need to go to the hospital and so. So use two kinase inhibitors. Second issue was it is difficult to combine three drugs at the same time. Why? Because when we combine three drugs, we also combine the side effects, the toxicities. Try to get down to two drugs. So we worked hard, and we found, we started working with the AXL kinase inhibitor. Turned out that the AXL kinase inhibitor, also EGFR inhibitor, induces reactive oxygen species that Yossi Shilo told you before. The antibodies do not induce the reactive oxygen species, as you can see here, for example. And this is osimertinib or the anti-AXL kinase inhibitor. So there is something very different when we apply a kinase inhibitor, two kinase inhibitors, or two monoclonal antibodies. In the next step, we tried them, the kinase inhibitors, two kinase inhibitor or two monoclonal antibodies in vivo by dual combination or triple combination. What I show you here is the dual kinase inhibitors plus one antibody, anti-EGF receptor. And you can see after inhibition, the tumors relapse. But when we try two monoclonal antibodies and a kinase inhibitor to EGF receptor, tumors do not relapse, and you can see the survival. Unlike the combination of two kinase inhibitors, to remind you, they induce ROS, reactive oxygen species, and probably accelerate up regulation of mutations. So remember the other demand from the pharma companies. They said, go ahead and try to reduce from triple combination just to dual combination of drugs. So as I write here, because the combination comprising two antibodies worked better in terms of relapses than the two kinase inhibitors, we decided to make a bispecific antibody, one arm to EGF receptor and the other one to AXL, replacing the two antibodies. And so it is shown here. This is cetuximab, anti-EGF receptor, approved in the clinic. This is antibody 654 that we made in mice. And this is a bispecific to EGFR and AXL. And this is what we obtained when we looked in mice. So here, these are individual mice, and the growth curve and the relapses. The most successful with the combination with the bispecific. You can see it here. These are the bispecific or the triple combination, the combination that the pharma do not really like. So when we combine two antibodies or the bispecific, we see about the same effect. I must restrict it because when we engineer a bispecific, it is my last slide. When we engineer the bispecific antibody, we always lose some affinity. We always lose some affinity, and maybe we should re-engineer them with specific mutation. So that is what we obtain, and you can see they are functionally equivalent. So these are my conclusion. Upregulation of AXL in response to death of neighboring cells because of the phosphatidylserine and sensing of apoptosis enhances persistent tolerance to kinase inhibitors. AXL activation initiates the SOS response, the one that was discovered in bacteria, but we showed that it is relevant to mammalian cells, cancer cells, the upregulation of mutagenic processes. When this was introduced in the first time, people do not really believe that there is a way for a cell, bacteria or eukaryotic cell, to introduce mutation from within. No UV like in melanoma, no smoking like in lung cancer. It is mutation coming from within in order to survive the stress. Also, treatment combining a kinase inhibitor and a bispecific AXL EGFR antibody can persistently prevent or delay, I should say, relapses of EGFR mutated models. So these are the people who did the work. This is a Hanukkah party in my home. And I want to thank Ashish Noronha and Arturo. and also Moshe Linton, who engineered the antibodies. Thank you very much for your attention.
Thank you, Yossi. Beautiful demonstration of how complex cancer is and how we need to attack from different angles. I'm wondering if you thought of doing a tri-specific antibody to the 3 targets. Is that a possibility?
Yes, we call it a monster.
A monster.
We call it a monster. This is cumbersome. We see reduction in affinities. We are still.
But there are 2 successful demonstrations of tri, so it is a possibility.
It needs more work.
Yeah. Questions? Tell me. Thank you. Do you think that this is the mechanism of the inducing of persistent cells in tumor? So, because there is a drug specific, and you are showing kind of on this direction. So, do you think that this work breaks one of the mechanisms of making them?
Yes, I think so. Maybe also melanoma with BRAF inhibitors and some MAP kinase MEK and so. I think so. I'm not the only one who thinks so. There are indications that indeed there is what we call drug-induced mutagenesis or treatment-induced mutagenesis, because our drugs now are so effective. They induce so much cell death. It's very similar to antibiotic and bacteria. So cells have the ways, and SOS is the last way, the last escape way. So it was shown, and of course there are variations, but it was shown in several systems.
Thank you. Thank you, Yossi. We'll continue. Our next speaker is Professor Naama Geva-Zatorsky from the Technion, who will speak about functional plasticity in the gut microbiome and its potential in cancer.
So thank you very much, Ronit, for inviting and all the organizers, and I'm really honored to be here. And it's has been a fun morning, and I'm really enjoying. So thank you very much. So and also thank you, Yossi, because it's exactly what I'm going to talk about: how the bacteria can, from within, change their functions, and that's actually one of the reasons I wake up in the morning. So I'm very excited about exactly this. So the microbiome, switching gears, but smoothly, thanks to Yossi. So the microbiome is a field that has risen in the past 2 decades, plus minus. It started by correlations between the microbes and diseases, but we are in the past decade even, I would say, starting to look into causation, and there are many really nice studies on causations. And we're really looking into not only the taxonomy of the bacteria, what are their names and how do they change, but I really encourage to look into their functions, like not only who are they, but what are they doing. So in this image, it's an illustration of one of our studies, of my postdoc studies, showing how the bacteria can actually play with the immune system and change the functions also of the immune system. So the gut microbes modulating the host immune system. So that was in my postdoc in Dennis Kasper's lab in collaboration with Christophe Benoit and Diane Mattis and Essen Sefik. So the microbes have a taxonomy. They do have the names: kingdom, phylum, class, order, family, genus, and species. And we see that the taxonomy doesn't relate, or we cannot tell from their taxonomy what will they do. So it's not that this taxonomy upregulates regulatory T cells and that one upregulates Th17s, but they are really merging. So we have to look at their functions and not only their names. But we also see now that the strain also matters. So me and Ruti can have the same bacteria, and probably we do share some bacteria with our close relationship. But they probably have also different functions because they are with their ecosystem, with other neighbors. We have other ecosystems of our own. So also the strain really matters. So you can see here the blue and the red. It's two different strains of the same species. So really the same name of bacteria, but they do different things. One upregulates the ILC3s in the colon and the other downregulates them. And in this correlation clustering, you can see that it's a study, computational study that Haitham Hajjod has done in my lab. And you can see the colors. So the two yellow ones show two strains of the same species that are similar to each other in relation to what they do to the immune system, but they are very different from another yellow, or you can look at the two pink ones, the one at the very edge, and well, I can show it here, this pink and this pink. They are two strains of the same species, but they modulate the immune system very differently. So the strain also really matters. But now, if that wasn't complex enough, we're interested within the strain. So what are the functions of the bacteria that can alter in response to the environment within a strain? Are they always the same? Can they change? And what alters their functionalities? So we're looking at the neighboring bacteria, the growth conditions, the host physiology state, which I will expand upon, bacteriophages. So the colors here are showing an illustration of the functions of the bacteria in response to the environment. We're also looking at their molecules and delivery platforms. So these are extracellular vesicles so that bacteria can secrete. And Noah has characterized these extracellular vesicles and also their proteomics, what are their molecular structures. And now an unpublished study that we're currently resubmitting, actually. She's showing how their functions and how their proteomic infrastructure is altered in different conditions and how does that affect the immune system. So that will come soon, I hope. So within the same strain, individual bacteria, are they always the same? Can they change? What causes these changes? And is there information in this variability? Can we measure this variability within the strain? And does it tell us anything about their prosperity, their stress? So we call it functional plasticity, alterations in the functions of the bacteria in response to the environment. And in other words, bacteria are complex and they have many different functions. I think that point was raised. And we mainly look at how the bacteria modulate the immune system. So in one case, a certain bacteria from the same strain upregulates an immune cell, something changes in the environment, something changes in the bacteria, and then it downregulates the same immune cell or affects another one. And this is a reversible phenomenon. So one of the mechanisms that can allow the bacteria to do so is DNA inversions in the genome, meaning that regions in the genome can flip and flip back in a reversible manner, depends on the environment. So in this case, and actually the simplest case is promoters turning on and off, but there are also more complex systems like Nadav Vanessa has shown here in collaboration with Laurie Comstock that are more complex systems in the genome of the bacteria that alter and change even global machineries in the bacteria. So this is an example. We didn't invent this. This is an example of a pathogenic E. coli from 1985 with a promoter that can flip on and off, and it alters its outer surface fimbriae. So in the case where the promoter is on, it has the fimbriae, and it can adhere to bladder cells, and this can be reversible. So when it's off, the E. coli is less pathogenic. And in the gut microbiome, it's only recently has been more and more gaining interest. Well, I would say 2001, Laurie Comstock is interested in phase variation for long, and several other studies as well. But in the past few years, it has gained even more interest in more global and complex system detection, mechanistic insights. How can we analyze the functions? And we are recently studying the relevance to the host state. So I will first talk about a study we've done on these functional plasticities. It's DNA inversion in the genome and phase variation in the molecular level. That was led in my lab by Shaked, together with Rawan and Haitham and Tal, my lab manager, and we collaborated with Itai Sharon from Migal on part of this study. So we analyzed Crohn's and colitis patients. So I'm saying it up front, but we have also studies related to this in cancer that I'll talk about in a moment. So we've analyzed cohorts of patients that others have done the metagenomic sequences of the bacteria already. And we apply another algorithm that can analyze these inversions in the genomes of the bacteria. So the algorithm we used here was developed by Eric Alman and Ramnik Xavier, by Jiang, and it's called Phase Finder, where it identifies the inverted repeats that allows the invertases to flip the DNA and flip back. So we identify the inverted repeats, then we simulate the forward and the reverse orientations, and we analyze how many forward we have, how many reverse we have, and in the different conditions. So these are phase variable regions. We identify the phase variable regions in Crohn's, colitis, and healthy patients from 6 different publicly available cohorts worldwide. So this is the heat map of the ulcerative colitis, Crohn's disease, and healthy patients. And the colors show the percent of reverse. So you can see that the patterns are different. It's the same bacteria, that same type of bacteria, same species of the bacteria, and even the same strain of bacteria in the different patients, but it has a reverse. Something is different in these orientations and the genomes of the bacteria. So the colors show these patterns. So we find across phylogeny, we looked mainly in the Bacteroides genus, but we saw also in other bacteria as well. So this is an example of Bacteroides tetaiota omicron, where we can see these flips. So the y-axis is the reverse orientation. So how much of the genome is on the forward or the reverse orientation. And these are CPSs, capsular polysaccharides. So the outer surface of the bacteria is altered. Blue is healthy, red Crohn's, yellow colitis. So you can see that there are significant alterations in the same bacteria in healthy versus diseased patients. Many of these alterations are in the outer surface. So we can assume that this relates or can affect the interactions of the bacteria, both between themselves, but also with the host and the host immune system. And this is another example that we further delved into, which is Bacteroides fragilis, also polysaccharides, where polysaccharides H and polysaccharides A were flipped in the diseased patients versus healthy controls. So these switches changed the outer surface. And polysaccharides A was actually studied very thoroughly by my postdoctoral mentor, Dennis Kasper. And this is a study by Sarkis Mazmanian when he was a postdoc in his lab, showing how polysaccharides A of B fragilis can reduce inflammation in the gut. So when polysaccharide A is expressed on B fragilis, the inflammation is markedly reduced in experimental colitis in mice. And what we see is in patients with inflammation, the promoter of this polysaccharide that can reduce the inflammation is actually turned off. So we asked what is the egg, chicken and egg, and whether it is a dynamic process. So to do that, we studied mice where we added the DSS for 9 days and then switched to water. And we see here, it's again the ratio of on orientation, because in PSA we know what turns it's on. So at the beginning, no difference between the mice that drank water and to the mice that received DSS. Then the mice that received DSS, PSA promoter was turned off. And after we removed the DSS and switched to water, it was turned on again. And again, there is no significance between the healthy and the DSS drinking mice in the past. And this was correlated with calprotectin. So first the PSA was turned off. Actually, it preceded the calprotectin levels. And then the calprotectin was on. So inflammation was induced in these mice. And we think it is kind of, we started to think that it's a response to inflammation. And you can see here also the body weight that was reduced in the DSS drinking mice. So we received, to study what causes this flip, we received stool samples from patients from the Rambam Hospital, from Yuda Chovers and Sigal Pressman, before and after treatment of anti-TNF. So we got the stool, we created a soup of the stool, filtrate without the bacteria themselves. We filtered the stool soup, and we applied it to this bacteria, to Bacteroides fragilis. And we see that when we apply this filtrate before treatment, PSA is off. So the diseased calprotectin on diseased patients, PSA is off, but after treatment, it is turned on. So it's something in this filtrate that causes this flip. What can it be in this environment? Probably you have more ideas than in this slide, but immunological imbalance, disease-associated bacterial alterations, bacteriophages or viruses and metabolites, biochemical shifts. On all of these, there is literature showing these changes during inflammation. We chose to look into bacteriophages. So we first went back to the cohorts and analyzed whether the bacteriophages are correlated with the PSA off state in patients, and found specific, high correlation of bacteriophages specific to B. fragilis in patients where the PSA was turned off. And these are, interestingly, bacteriophages that were shown by Nishiyama et al. That are associated to active disease. And you can see here that they are active because their ratio is higher. So they are proliferating, and the bacteria are more in the off orientation of PSA. So we decided to isolate a bacteriophage, and Nadav Benassa became an expert of isolating bacteriophages. So you can see here the plaques. It's a bacteriophage against B. fragilis. Actually, the one I'm going to talk about is not the one Nadav isolated, but still he's wonderful in it. It was isolated by Joan Joffre in Barcelona. So it's a lytic bacteriophage. It's a C4-VDA, and it is specific to B. fragilis, and it's actually in its genome very similar to the ones we saw in the cohorts of the patients. And so we asked, okay, we have this phage in our hands. Let's see if it can actually induce the DNA inversion. And we indeed see that in mice, the PSA is turned off when we give the bacteriophage. The mRNA of the first biosynthesis loci of PSA is also turned off, is not expressed. And also the polysaccharide is not present in the capsule of the bacteria. B. fragilis with PSA reduces inflammation via upregulation of regulatory T cells. So we asked whether the bacteriophage that we already saw that can reduce the polysaccharide A, does it affect the B-Frag Treg interaction? And we indeed see that when we add the bacteriophage, the regulatory T cells are significantly reduced. So it has actually an immune effect on the host. So this was published, and we were honored to be included in the Pi Day in cell host microbes, saying that we added a piece to the microbiome Pi, looking at these DNA inversions, and indeed now we're looking at it in many different physiological states, including cancer, and we have some really nice and strong results in collaboration with MD Anderson, with Jennifer McQuaid and Jennifer Vargo and team. And Nature Reviews Gastroenterology have chosen us as one of 3 studies that were influential on IBD in 2024. They did a really nice graphical abstract. Always others do your graphical abstract best. So microbiome has been added to the cancer. This is not us. It's others have shown deleterious and beneficial effects of microbes, and we have several studies now on esophagus cancer, where we see that the functions of the bacteria in the esophagus tumor have relation to oxidative stress, and it relates to anastomotic leak, the surgery that is given to esophagus patients, and that's with Irit Ben Aaron, unpublished. And also in Lynch we saw correlations, but I see that I don't have any more time. So DNA inversions of microbes can be potential biomarkers. They can also have potential effects on therapeutics, as we saw with the regulatory T cells, and we now see also in the cancer patients. And I encourage us all to look not only on composition, but also on microbe-host interactions, on the functionality and functional plasticity. And thanking the lab, the funders, and you for listening. Thank you.
Thank you, Naama, for adding this dimension to the hallmarks of cancer. Any question? One.
Beautiful talk, Naama. So when you did the DSS treatment, it was one cycle, right?
Yes.
And if you repeat the cycles, do they undergo this flipping again? So do you think there's a difference between acute and chronic inflammation as an implication?
It's a great question, and actually, it's a great question, and we didn't do that. And it reminds me that I also wanted to say that in patients, I thought while preparing here to this talk that it would be really interesting to look at the patients with IBD that progress to cancer. You're asking in this also because of the reversibility, and also, I guess, because it can also lead to colitis-associated cancer. Yes. I'm intrigued by that as well. Thanks. Thank you.
One last, very urgent. Okay.
I was wondering if this inversion mechanism for general plasticity also exists in like mammalian genomes or maybe in humans?
Yeah.
I'll just repeat the question
yes.
because you need a microphone for the audience.
So the question was whether this inversion mechanism also exists in the mammalian cells. There are some related mechanisms in mammalian cells, but to me it's like well, I became a microbe. We microbes don't have exons, so I see this as a compensation mechanism for the variability that the mammalian cell has. Yeah.
Thank you, Naama.
Thank you.
Thank you. Our next speaker is Professor Rachela Popovtsa from Bar-Ilan University, speaking on the golden approach for brain metastases. Welcome.
So thank you very much. Thank you very much, Ronit, for inviting me, and it's wonderful seeing and meeting all these great people here. So what we are dealing now and doing in my lab is mainly trying to go over biobarriers with nanoparticles, and I'll show you one piece of our work today. So I think that we're all familiar with the blood-brain barrier challenge. This is a real challenge in which most of the drugs are not entered to the blood-brain barrier. We know that we have too many brain diseases, from brain cancer to Alzheimer's disease, Parkinson, many others, trauma in the brain, and we don't have so many drugs. Basically, we have today something like 130 antibodies that were approved, and only less than 3% of them were approved to the brain, and still they face many challenges. So not just going through the blood-brain barrier, usually this is the highlight, or what people refer to the main barrier, but also after going through the blood-brain barrier, we still remain with a challenge. There's a challenge not just to go through, but to reach to the exact place, to reach the desired organ or desired disease, and then to reach to inside the cells, whether it's in the periphery of the cell, inside the cells, in the cell microenvironment. And if you think to yourself, these are multi-scales biological challenges, from the whole body to the brain, to the specific location in the brain, and then to specific organ, which this makes this challenge really reinforced. the main strategies for delivering drugs to the brain are invasive techniques, in which there's a direct administration through the CSF, spinal cord, many invasive technique with their own advantages and disadvantages. And there's non-invasive technique, such as temporarily disruption of the BBB and pharmacological strategies. And the main strategies today are modification of antibodies, or modifying the antibody with some shuttles, and the transfer is the main shuttle that is being used today for transfer the BBB. And the other one is the nanoparticle. So for the people in the audience which are not familiar with the nanoparticles, so I'll just mention why and what is the main advantages of nanoparticles and why we use nanoparticles. So we all know drugs, and the drugs that drugs both exist today and developing today, they have their constant properties, meaning they have very defined pharmacokinetic, biodistribution, half-life time, and we cannot or can hardly change it. But while we are adding them somehow to the nanoparticles, we have a huge playground for modification. We can change mostly all part of its kinetics, pharmacokinetics, biodistribution, whatever. And if we play smart, hard, or be successful, so we can enhance the accumulation of the drug in the specific organ, in which we envision reduce the toxicity, and meaning that sometimes the toxicity of the drug is mainly focused in one organ, and while the nanoparticles does not reach specifically to this organ, we can reduce the toxicity. Sometimes we alter the toxicity, and to overcome biobarriers. We are specifically, we're using gold nanoparticles, and one more advantage to gold nanoparticles is that we are able to see them using a CT. And this is an advantage. This is the whole area of theranostic, which is interesting. But other than that, in clinical trials, the success rate of drugs, theranostic drugs, are a bit more than the regular drugs. So if it's one digit percentage of drugs to become clinically available, so the theranostic drugs are 2-digit. I guess it's not 99%, but something that close to something between 10 to 20%, because the main advantage is that we can stratify the patient before or at the beginning of the clinical trial. And just to show you why gold is so nice, so this is, for example, this is an image we did in my lab, a CT image of a mouse in this exact specification. We see only it's a CT, we see only the bones and no hard, soft tissue. But after IV administration of gold nanoparticles just to the blood, this is what we get. So we can see very nicely the very fine details of all the bloodstream, and this is because of the high Z number of the gold. We can very nicely see it on a CT. Our technology, so it's basically a platform in which we can conjugate to the nanoparticles many different stuff. So we have antibodies, we call it golden antibodies at different directions. We have bispecific antibodies, and it was nice to hear your talk before, because it was just, I'll show you later, our bispecific particles. ADC, we can conjugate, other than conjugating antibodies to the nanoparticles, we can add also chemotherapy, and I will not talk about it today, but basically we can go over, we have different challenges, but we can go over existing challenges of current ADC, like low DAR, antibody-drug ratio, and because we can conjugate multiple type of chemotherapy on the same particles, different type of chemotherapy on the same particles, and many others, and also to add radiotherapy to the nanoparticles. For the brain delivery platform, our strategy was to use an insulin. And we are using insulin because, so I'll start with the particle itself. So the particle, it's a gold particle. It's coated with different linkers. Insulin is our Trojan horse to go through the blood-brain barrier, and then therapeutic cargoes as antibodies. We have different lengths of, we are playing with this, with the linkers, because as I mentioned, we not just want to go to pass through the blood-brain barrier, but we also need to reach a specific location or specific disease within the brain. So some interesting fact on insulin, you can also read it as why we envisioned or that we can be as good as or better than the transferrin, because the transferrin is the main strategy in which the pharma companies are used today, is first of all, there's a very high overexpression on brain microvessels of insulin receptors, specificity compared to peripheral organs, and also the same insulin, or almost the same insulin, is transferable from all species. So the same insulin that we are detecting on mice models exists in humans and in monkeys. There's also a much greater availability of the insulin receptors on the BBB membrane, and usually it's unoccupied, as opposed, for example, to transferrin, in which 99% of the transferrin is constantly occupied by a native transferrin. And also insulin receptor and also insulin growth factor receptors, in which the insulin can bind to both of them, found to be highly overexpressed in various cancers. And this is also very important. So the first, just to show you, sorry, our ability by using a CT. So what we have here, we see the particles, the gold particles with insulin. We can see how nicely we can see the particles within the brain. This is without any disease. And without insulin, nothing gets into the brain. So the gold particles cannot get into the brain. So the insulin is actually our Trojan horse, in which we deliver, we can take him and deliver drugs to the brain. We found out that we are 40 times better than free gold nanoparticles, the free without the insulin, 30 times better than free antibodies, and 10 times better than transferrin and gold nanoparticles. This is another demonstration of, it's a super resolution microscopy, in which here you can see the blood vessels it's a slice of the brain, in which you can see here the brain artery, and at this time the antibodies are fluorescently labeled, and we can see them stuck within the artery, while using our gold nanoparticles, we see that how they diffuse into the brain itself. This is another example of a real-time in vivo two-photon microscopy. It has been done here in Tel Aviv University, and we see here again what we see fluorescently labeled is the antibodies and not the gold nanoparticles, and the free antibodies here are in the artery, and they are predeterminedly stayed within the artery, within the blood vessels, while here we can very nicely see 24 hours later the antibodies that are excreted from the blood vessels because of the insulin. Another demonstration that we have, this is trastuzumab. We have a free antibodies that we can see him. Basically, this is before injection. After injection, we can see how the blood vessels change his color, and the antibodies restricted within the blood vessels, while by using nanoparticles, we can see them, insulin-coated gold nanoparticles, we can see them excreted from the blood vessels. Here is another control in which we took gold nanoparticles with insulin and gold nanoparticles without insulin. So we see actually that the insulin itself, not the particles, not everything else, the insulin itself causes it to get out of the blood vessels. So we are now in the process of, we are doing a lot of research of understanding the mechanism of the insulin in not just passing through the blood-brain barrier and can get out of blood vessels, but also a different characteristic. We are only in the beginning, but we also saw that because of, or maybe because of, we see a lot of insulin receptors on different types of cancer, that the insulin itself can act as a targeting agent. And we see that without insulin, if we take here, it's with antibodies. If we take nanoparticles without with antibodies without insulin and with insulin, so the total uptake within the tumor is much higher when we use also the insulin. So the insulin also can act as a targeting agent on top of the antibody. And also we can see that the insulin itself, we can see here that the nanoparticles with the insulin can get really more deeper into the tumor and not just in the periphery. So now I've just, now I've just spoke generally on the insulin and our capability of the platform, but then this, we opened a company, a spin-off company of a startup company of my lab, and I'm a co-founder and a CSO of this company, in which we have chosen to use these nanoparticles to deliver drug to brain metastasis. And why we have chosen this model of HER2-positive breast cancer brain metastasis? Because the brain is a complicated issue, and today we have very, very few antibodies that treat neurodegenerative diseases, but we had to find a model in which the only problem, the only parameter that is missing is going through the blood-brain barrier, but the activity and efficiency of the antibody and the disease is well known. So all the neurodegenerative diseases went out of the game, and we have chosen the HER2-positive breast cancer because we know that Herceptin and Perjeta are well-known antibodies that were treated for many, many years and have proven to have the survival rate, and they proved to be highly effective. While with brain metastasis, these patients are treated very well, but once they have a brain metastasis, this is basically the end of the game because the antibody cannot enter the brain. So this was our model, and now I'm taking you a little bit to another story, is the bispecific antibodies. So a little bit, I guess that we also have some limitations, but other than trying to build a bispecific antibody from the beginning, we just take to our nanoparticles, mix together two types of antibodies. And since the treatment for brain metastases are trastuzumab and pertuzumab, we took these two antibodies, conjugate them to the same particles, and practically we have a bispecific particles, bispecific antibody, and we can do like this trispecific, four-specific, any monster that you envisioned. I guess there's also a disadvantage for this, but here I will show you that this actually worked very well. It's, we see here on cells, this is only on cell line, so we see that our bispecific particles are better than just adding free antibodies, this mixture together free. By using the particle, it's better than the free antibodies, and also we couldn't compare it to bispecific antibody because I don't know if now it exists, but it doesn't exist, and I don't think we can buy it. But we see that it's better also in two different cell lines of HER2-positive breast metastases. We can see that they work well. We also investigated the mechanism, and we see that if the free antibodies get to the cells within the mechanism of the HER2, we can see in our particles the same mechanism. We also saw that actually we have started with a problem, with a challenge. We couldn't, Danny Perry is not here, but he is the famous of successfully bind antibodies in a very specific orientation to the nanoparticles for targeting issues. So always the antibodies were very nicely facing out the Fab part was face out of the particles, and we could nicely target. We couldn't do it, or it wasn't easy to do it. So at the beginning, we thought, okay, we are losing some of the antibody. We can conjugate around 30 antibodies, and some of them are being lost. But then we realized that this way we are preserving or inducing the ADCC activity of the antibody, which is also very, very important. So we see here on a cell in vitro, we see that while adding NK cells, we can see that we have ADCC activity to the nanoparticles. And in vivo, so this is still in the periphery, it's not in the brain. So before we went to the brain, we wanted to check if in the periphery it works. So here we can see the controls. We see the HER2-positive breast cancer. This is without any treatment. This is treatment of the free antibodies, 2 types of antibodies. Here is a cocktail of our antibodies, meaning that half of the particles were conjugated with trastuzumab and the other half with pertuzumab. We just mixed them together, which was similar to the free antibody, as in very logical, because to the periphery, nanoparticles reach the tumor and free antibodies reach the tumor. But we saw that by doing the bispecific antibody, we got really better results. But now moving to the brain. So as I mentioned, it's very hard to get and to reach to a specific location. So this is a model of, this is a mice model of HER2-positive brain metastases. Wow. And we see that this is an ex vivo image of the tumor. This is iodine of, just to see where the tumor is. Here are our gold nanoparticles. So we see that the exact colocalization is exact on the same place of the tumor. And this is just a picture that we can see by our own eyes, the drug within the brain, which I think it's very impressive because it means that there's a huge amount of drug. This is actually the color of gold nanoparticles. It's something like a red wine, and we really nicely see it in the tumor. We also saw some good results, of course, with MRI. We have checked the tumor size throughout the experiment and saw that our particles are better than the free antibodies. Also, we see that we extend the survival. These are free antibodies. We could extend the survival by more than 50%. And basically, so what we have done, we've took the same animal model, same cells, same everything of the experiment in which they developed the trastuzumab pertuzumab. And then the previous development, which is being used, and the clinical trial of this was translated to 15 and something months. So we know that always the question is, how do we know that mice experiment will see it at the end, the same effect on humans? So here we are hoping, because the same effect that it has been demonstrated before in the periphery, now we saw it in the brain. So with this, I would like to acknowledge my group, NanoCarry, and the grant. Thank you very much.
Thank you. Thank you, Rachela. We have a question. Do you have the microphone? Just a second. Neta.
Super interesting, of course. I was wondering about the immunogenicity of the particles, because if I'm not mistaken, the experiments you showed are in nude mice. So I was wondering if you also did these experiments in immune-competent mice, and what did you see? Is the immune response, are they immunogenic? And if so, does the immune response actually is it helpful or not to the activity of the antibodies in brain metastases?
So basically, I think that this is thanks to the part of this research was done in the company, that they have much more money than the university to do many toxicity studies of different types of animals and different time point and everything. So basically, the gold itself is not toxic in any way. And we don't see any alteration of any kind of toxic response. The only thing is we are just following all the effect of the antibodies themselves. We didn't see any increase in immunogenicity at all.
Last question, Asaf.
On the same lines, so the gold is there to stay?
No, of course not. Of course not. The gold excreted from the whole body, actually from the brain even much faster than other organs. The most problematic organs are the liver and spleen. It could stay in the liver up to 2 months, but from the brain and other organs, it gets secreted very fast, meaning hours. Other than sometimes when it stays more, when it's conjugate, for example, when you target it with a specific antibody or something like that, it could stay more, but it's secreted very nicely. Nothing remains in the body. We are not developing the next golden person.
Yeah. Goldfinger.
Yes.
Thank you, Rachela.
Thank you.
Our next speaker, back to the clinic, Professor Talia Golan from Sheba Medical Center, will speak about therapeutic exploitation of DNA damage deficiency in pancreatic cancer. Thank you, Talia.
So I'll start saying that I'm a physician scientist, and in that order and in that priority. And therefore, it's a huge privilege for me to be here and to speak in with such scientists.
Wait, I don't find you.
Here I am, yeah. So I'm going to speak specifically about homologous recombination deficiency in pancreatic cancer.
Do full screen.
There we go.
Yeah.
Okay. And really a small introduction on pancreatic cancer to give you the context of BRCA-associated pancreatic cancer, and then about the prognostic predictive value of a BRCA-associated pancreatic cancer, the model systems that we have tried to develop to recapitulate the specific subset of patients, and then resistance mechanisms and maybe the future in the subset. So why? We don't have to ask me why, because it is all I really think about and dream about. But pancreatic cancer should bother us all. In all the solid tumors, it's the one tumor that is still extending in its prevalence, and we haven't impacted mortality almost at all. This is a very humble slide that shows the therapeutics in pancreatic cancer in the last 25 years. This is a very, very limited slide. It shows you can see the chemotherapy and then the biological agents, and you can see that this is a very limited slide if you compare it to lung cancer, to breast cancer, to melanoma. So we've got a lot to still do in this field. This shows you that there's a significant unmet need to really disease control. And this really gives you the subtext of what we're dealing with. Most of our patients, only 15% have a local disease. That means the rest of those patients have advanced metastatic disease. And this is a progression-free survival in first-line setting. This is really limited. And very few of our patients even get to second-line setting. So we looked at the outliers. Those patients with short and long-term survival in pancreatic cancer. This is the real-life data, and this is even more depressing. One can see that, yes, patients who live for more than a year, the prevalence of those patients, a small percentage, are improving over the course of time, but one can see that those patients with very dismal disease that are dying within 8 weeks of diagnosis is really not changing, and this is a very huge amount of our patients, about 40–50% of our patients. So this is really the bread and butter of what we deal with. And in that context, I'm diving into germline BRCA-associated pancreatic cancer. This is about 4–7% of patients with pancreatic cancer. BRCA1 and 2 are key proteins in the homologous recombination deficiency pathway. And we know that when we look at their genomes, they show unstable genomes. I'll show you a little bit soon more. BRCA is a predictive biomarker. It is not a prognostic biomarker. So if we do not identify these patients, if we do not tailor our treatment to this specific mutation, then they have the same prognosis as the other patients. I'll show you some data on this. And we have others have showed that PARP inhibition in the maintenance setting can be beneficial for these patients. So this is retrospective data. And here you can see that if you do not give these patients platinum agents, okay, the overall survival is the same as the regular patients. So we can say that this is more a predictive biomarker than a prognostic biomarker. But where can we go with platinum-based treatment? This is a picture I show from a very dear patient of mine who passed away. She was 26 years old. She had a BRCA mutation in pancreatic cancer. She had a very beautiful, deep response to FOLFIRINOX, which is combination treatment including platinum. But when I came to see her in the ward, I said to her, you know, why is the chemotherapy covered with all these beautiful scarves? And she says, it is causing me such terrible nausea that I can't even look at the chemotherapy, so I'm trying to cover it. So I said to her, I'm going to take a picture, and I'm going to put this picture up, and we're going to remember where we can get with chemotherapy and how much more we have to do for our patients. So this is also in her memory and in memory of many other patients. And this really leads to PARP inhibition. So the synthetic lethality that is shown with PARP inhibition in patients with BRCA 1 or 2 mutations is already well recognized and well known, especially to this audience. And we actually, together with others, developed the phase 3 clinical trial in which we gave PARP inhibition as maintenance after exposing the patients to 4 to 5 months at least to platinum-based chemotherapy. One can see a nice progression-free survival. So here it's about 7.4, so this is plus at least 4 or 5 months. So we're going on to over a year progression-free survival. Remember that second or third slide I showed you with a very small limited progression-free survival. So I agree that this isn't good enough yet, but we're already starting to push the envelope, okay? So we're starting to see a signal. Overall survival, which was our secondary endpoint, was unfortunately not statistically significant. But once again, here we can see a trend. And before I go deeper again into BRCA-associated pancreatic cancer, I do want to give you just a bit of a feeling about HRD in pancreatic cancer, because I think it is relevant. So we, together with others, specifically at OICR, they have whole genome sequencing, which they developed through microdissection capture. So the cohort is very clean. We looked at about 400 patients with pancreatic cancer, and we asked a very simple question. Who has HRD besides germline BRCA? So how did we define HRD? We looked at known classifiers, which included the instability score used by Myriad, the substitution-based signature 3. This is Alexandra's Nature paper from 2013, the HRD detect, another score, and structural variant burden. And when we looked at over 400 patients, you can see here the patients that show an HRD. So that's in the blue rectangle. Those are all the patients that showed the HRD classification. And you can already see here that there are some patients in the red box that have germline BRCA mutation. They're HR proficient. They aren't HR deficient. So we're seeing that when you look at the genomics, not all the patients will show the classifier as we think. And this slide summarizes that study. Here we can see that the majority of the patients had a HR proficient tumor. And then we start seeing who the patients who have a HR deficient. So those with germline BRCA1, germline BRCA2, somatic BRCA1 and 2, PELP-B2 and RAD51. And you can see here that all the other classical HR genes like ATM or CHEK2, none of them showed the HR deficient score as we defined it. Another very interesting group was the tandem duplicator phenotype. They are enriched with patients with rearrangement signature 5, increased number of small deletions, and some of them only have a BRCA1 and 2 mutation, and the rest of them don't. So you can only define these patients based by whole genome sequencing. Now I'm going to dive back into germline BRCA mutations. Okay, so what you've seen that it's a predictive biomarker. You've seen the phase 3 study. But what can we really speak about when you think about the spectrum of response? So here I'm showing you 100 patients treated at Sheba Medical Center over the course of about a decade. And it's very easy to see that you can see a spectrum. Okay? There's a spectrum here of patients. At the bottom, you can see what we call the refractory patients. So even though they have a germline BRCA mutation, we expected them to respond to platinum-based treatment, or a PARP, they did not. They are refractory. They have limited overall survival. Then you have at the top the long-term responders. Almost 10% of our patients who go into long-term remission. For pancreatic cancer, that is remarkable. It gives wind to our sails. It makes us know that here there is some patients you could tease out a very significant response. They live for over 10 years, some of them, and they're in remission. But the bulk of our patients, which we always concentrate on, are those that have a beautiful response to platinum-based treatment, and then they receive their PARP inhibitors, and then they go on to develop resistance. So they have an acquired resistance phenomena. And now we try to think how we could model these patients and learn about them, so really from the clinic into the lab. So over the years, we've developed the traditional PDX models. We've got today about 160 PDX models from different patients over the course of time, and we have about 35 of them that are BRCA associated. Okay, so we, like vouchers, kept on going for more and more. Just a small, I used to be very embarrassed to ask the patients to give us diagnostic and research biopsies. I used to feel that maybe we're exploiting them in a certain way, but today I look completely differently at this. I explain to them what we're doing, why we're doing this, and why they should participate. And I think that this is really a universal maybe message, that this is really all our problem. It's not one clinician or one scientist problem. So here you can see these three different spectrums that I discussed earlier. So the refractory, the models, the mouse models that will be like the refractory patients, those that will have the acquired resistance, and those that will be the super responders. Okay. So now we've got the mouse models. Now we have to show that these mouse models really recapitulate what we are seeing in the patient. So you can see the clinical subgroup. So in pink, you can see the acquired resistance. Those are the refractory and those are sensitive. And you can see that it's color-coded. So if we said that the mouse was developed from a patient with acquired resistance, when we gave in vivo or in vitro platinum or PARP, you can see that our mouse models reflect the clinical subgroup. So we think that they really do recapitulate what we're seeing, and now we can start analyzing these model systems. The problem with PDX is known to this audience. I don't need to expand. So we work together with Ravid Strassman and his lab on the ex vivo, the EVOC model system. So this is, you take fresh tumor, you put it into a special condition media, and you can grow it for several days, and you have cells that are live, and then you can give them treatment, and you can analyze the response. So what we can show here is that when we looked at the ex vivo culture system, and we correlated to how our PDX models and the in vivo results and the patient's clinical response, we now have a model. We have three models. We have two model systems, if you include also the culture, so three model systems that actually all give a very sensitive and accurate prediction of the patient's clinical response. So now we can use these model systems also to analyze, okay? We can't always just predict that what we've made works. So what are the mechanisms of response and resistance? Now we're getting to the more interesting things. So look at BRCA allelic status. So this is very basic, but still I'm going to just remind us all. So if you look on the monoallelic tumors, that's on your right, you can see that even though you can have a BRCA germline mutation, in the tumor you can still not have a somatic mutation, okay? So these are monoallelic tumors. However, which we have a biallelic tumor, that means that you can have or loss of heterozygosity of the second allele, or you can have biallelic inactivation of the second allele, and this could cause the biallelic tumors. And obviously, the patients that have the biallelic tumors they have a more chance responding to the platinum PARP strategy versus those with the monoallelic tumors. And when we looked at clinical response, refractory or acquired resistance, you can see obviously that there's a predominance for biallelic inactivation and response to treatment. So this is assumed, it makes sense, but it needs to be shown and proven. What about additional mechanisms of resistance? So this is a patient of ours that came in with pancreatic cancer, very aggressive treatment, very aggressive tumor. We gave him platinum-based chemo, and then he went on to receive PARP inhibitor. And then he developed resistance, and he developed ascites, and this is about almost a 3-year period. And from his ascites fluid, we took the tumor, we developed the PDX model, we sent the PDX model for sequencing, for whole genome sequencing, and what you can see is that he developed a frame shift, okay? So his secondary mutation that he developed was a reversion back to the reading frame, and this is how he managed to, from being an HR deficient tumor, he became an HR proficient tumor. So what do we know about reversion mutations? So secondary reversion mutations, we saw them about in 30% of our cohort. Is this a true prevalence? And this really brings me back to something that I think Shai today said. Frontal, temporal ignorance can lead to scientific catastrophe, and I'm probably not saying it exactly, but what I think we're trying to say is that you can always look and think of what you're looking at and how that extends the true signal. But we've got to remember that we're looking at a sample from one patient, and we're looking at one sample from that one patient. We're not doing a post-mortem. We're not taking all his tumors. We're not dissecting all those tumors. So we've got a very small, limited response. So I imagine that the 30% that we've seen is not reflective. We imagine that it's more than that. I don't know if it's 50, I don't know if it's 70, but it's more than that. And we've got to remember how limited we are in what we're looking at and what questions we're asking and what we're diagnosing. We've also taken the resistant tumors and done whole genome sequencing. And what you can see is that that in the patients with the secondary mutations, they're there as the HRD, so they were HR deficient, and then they developed the secondary mutations, these patients. You can see that they're the highest TMB, tumor mutation burden. Kind of makes sense, but it's not quite sure why. Maybe this reflects a little bit what Ruti said earlier. Maybe the higher CAFs that are immunophenotype, there may be something interacting with this that we're seeing, and we'll get back to that later. Another model that we've used is what we call the acquired resistance model. So here we take a tumor, we give it platinum, and then we give it PARP inhibition, and then we do whole genome sequencing. So what you can see that there's really a high concordance between the SNVs and the allelic frequency in the sensitive and acquired resistant samples. However, when we look specifically at the BRCA2 mutation, so the wild type allele, as the tumor became more resistant, the wild type allele grew. So something that we're really able here to model and to show, and it was the first time that we've been able, or any of the groups in pancreatic cancer have been able to show. So once again, it's intuitive, but you still have to prove it, right? So we don't know how many of the patients that we are treating are actually, we know that there's some kind of, preference, and we're teasing out the wild type alleles, but we're not sure what the preference of those is. It's still a technical thing in detecting these alleles in these patients. And this is a kind of summary slide before I go into the future. So this really shows a small group of patients, the ones at the bottom in the red, so there's about 5 of them. So even though they had biallelic innovation, they did not have reversion mutations, they still did not respond. So what's happening to those patients? And here we did some nice collaborative work with Uri Ben David and his group here, and you can see that, first of all, when you look at these patients, they have basal-like subtype, which is a more aggressive subtype, and they're more aneuploidy. So you can start thinking now about things that are directly related to the BRCA gene, which I would call intrinsic to the BRCA gene, like the allelic, the wild type allele, or reversion mutations, things that we can show that are directly related, things that are extrinsic to the BRCA gene, but are influencing the tumor, maybe aneuploidy, maybe the basal-like. So starting to tease out how we think about things that are going to be intrinsic and things that will be more maybe host-related or alternative pathways related, etc. We don't know what makes the patients long-term responders, but there's a variety of things that we're looking at to try and understand these patients more. I can just tell you as a clinician, I can almost smell these patients out where they come in. There's something about them that I can There's something in the host, there's something in the psychological impact, there's something that interplays between them that sometimes in some of these patients also reflects into a long-term responder. So what can we do to extend response or delay or overcome resistance? Maybe that's a more realistic approach. So we could try and mold the evolution of the cancer so that it's still inevitable that resistance is going to be developed, but maybe we can try and get ahead of the tumor biology. For example, what we've spoken about, things that are intrinsic to the pathway and then things that are going to be intrinsic and how we think about that. We can try use combination or sequential treatment to target emerging resistance. And the biggest challenge seems to be the reversion mutations, but maybe we can also try and target the chromosomal instability a little bit, what you spoke about earlier this morning, Yossi, and think about how we can bring all those things in and, of course, into what sequence would be nice to know. So this is going to be my last slide because I don't think I've got more time. But I just want to, obviously, you can do for how can we extend. So obviously, targeting alternative DDA pathways, WEE1, et cetera, that's kind of obvious. Immunotherapy, what you spoke about, the CAPs that are more immunophenotype, and we've showed a higher TMB. But what about the dual targeting oncogenic pathways? And here I'm going to cite you, Yossi, because I haven't used it before. So he spoke about centrality and lethality principle. Okay, so we have the KRAS mutations that are central. We have the BRCA mutations that are central to the tumor, both key mutations driving mutations, and how we can use those two as a dual target for oncogenic pathways. And we're starting to work in that, especially in the area now of KRAS inhibitions, and it's a very exciting time, I think, for pancreatic cancer. I'm going to skip this because I don't want to speak quickly. I'm anyway speaking quickly, and this just summarizes. And before I finish, as I started, I'm a physician and then a scientist, and most of this work's done by my team. I have a fabulous lab. I have no words to, and I think any of you in the audience here, I've met my lab, know exactly what I'm talking about. So Dikla, Khani, Maria, and all the team that are phenomenal and really take these clinical insights that I see on my patients and I bring back to our lab, and they can speak about the patients and they can speak about the translational research the same as I can, and I think that that's really the strength of being a physician that works in academic center, can also try and do research. So once again, thank you very much.
Thank you, Talia, especially for demonstrating once again how we can take from bedside to bench and back. So beautiful demonstration. Questions?
Beautiful talk, Talia. Maybe I missed this, but do you see a difference between, in that slide that you showed of the non-responders at the end, do you see a difference between BRCA1 and BRCA2 in terms of these long-term responders and the biallelic
Yeah, the numbers
switches?
are very small. There's more BRCA1s in the non-responders, but the numbers are very small. In our phase 3 study, so we tease the patients out. Only those responding to platinum went into the trial. So we don't see their difference. If you have a tumor that responds, it doesn't matter if you're BRCA1 or BRCA2, you get the same prognosis. However, it seems to the BRCA1 seems to be a little bit more aggressive subtype.
Thank you. We'll move to the last speaker. Our last speaker is Professor Oded Charan from Tel Aviv University, who will speak on data integration in a network context. Thank you.
Thank you for the opportunity to present here some of the works in my lab on integrating and modeling biological networks. Specifically, I'd like to present today two works. The first, BERT work by my student Rami Nasser, is on integrating different omic data sets. And the second, by my former student Daniel Pirak, is on learning directions of signaling flow in a physical network. So I'll start with the first. So the challenge is as follows. We are given multiple data sets. They could be either genuine networks, or they could be simple omic data sets that are represented as networks, for example, co-expression or collocation. And the goal is to somehow integrate this information together in order to learn predictive gene representations that can be used for any other task, for example, function prediction, module detection, etc. And the state of the art in this regard is a method that is called Bionic, which learns a representation from each network by itself and then in some ad hoc manner averages those representations to get the final representation of genes. And when we read this method, we were not satisfied with the way the integration is done, and so we thought about the following process, which we call BERT walk. And the idea is as follows. We operate in 2 stages. In the first stage, we take each of the networks and transform it into a document. And the way we do it is as follows. We think of any gene or any node in the network as a word. And then we perform random walks in the networks, and each such walk, we think of it as a sentence. And now, after we have these documents, the integration is merely done by merging all these documents together and feeding them to a large language model. And what this large language model knows to do is to learn a representation of the words, which are the genes in our case, in a contextual manner, which means that any gene knows in its representation about nearby genes in the network. And so we learn this representation, and then we use it for different tasks. So some benchmarks and sanity check that we do are adapted from previous work. So we apply our method first to networks from yeast, so 3 different networks, protein-protein interaction, genetic, and co-expression. And later we also apply it to human networks, again, PPI, genetic, and co-expression. And in order to evaluate the representations, we check how well they can reconstruct known modules or functions obtained from databases such as Intact, KEGG, Go, and Co. And so as a sanity check, we applied our integrated representation to predict both modules and gene function and compared it to learning a representation from each network individually. And as you can see here, the integrated representation outperforms each of the individual networks in all different standards and also on both prediction tasks. So this was a good sanity check that shows the power of integration. And next, what we did was to evaluate our method against previous work, again on the same standards. We did it first in yeast, and then also in human. And again, otherwise I wouldn't be standing here. In all cases, our method could outperform the state of the art. Now, what is specifically interesting in this large language modeling of networks is that the large language model allows us to learn not only a representation of single genes, but also of entire walks. Because of the way it represents sentences, if you're familiar, as tokens. And so what we did was to check whether this new representation of a walk rather than a single gene can help us to predict real pathways in the network. And so to check that, what we did was as follows. We synthesized many paths or many walks in the yeast network. And then we used knockout gene expression data from yeast in order to identify cases in which a gene was knocked out, and as a result, another gene changed its expression level. And so we defined a pathway, a walk in the network to be a real pathway if it started with a knockout gene and ended with a gene that changed its expression, with a differentially expressed genes. And we wanted to see whether our representation can detect such pathways as opposed to simple random walks. And indeed, we could see that our approach was much more powerful in detecting real pathways compared to a layman benchmark in which we simply average the representations of each of the genes across this walk. So this shows you the extra power of this modeling approach that allows you to represent not only single genes, but also whole pathways. So now switching gears and moving to my other work, as you know, if you take a physical network, when you measure it experimentally, you can detect only whether there is a connection between two proteins. But in fact, about 2/3 of these protein-protein interactions are naturally directed. For example, from a kinase to its substrate. But of course, the problem is that we cannot experimentally, in large scale, measure these directions. And so the question is, how can we attempt to learn these directions computationally? And this is very important because if we would like to go beyond our topological description of networks to something that is more logical or functional, we need on the way to reveal those directions in the network. And so the way we approach is as follows. We use indirect information from perturbation experiments, for example, from knockout experiments. And the idea is as follows. If you observe that when you perturb a gene S, you see an effect on gene T, then we hypothesize that there must be a directed path in the network that connects S to T. Now, this gives a result to many beautiful combinatorial problems. However, all these problems turn out to be computationally hard, and if you insist on solving them, then usually what you get is a very small coverage of the interaction. Somehow these methods are focusing on edges that lie on shortest path, and most of the edges in the network do not lie on this path. And so about 5 years ago, we came up with a method with full coverage that is local in nature, and I'd like to show you this method in order to introduce what I'll show next. So the method is as follows. So here what you can see two pathways that go from left to right, but all the directions are hidden, okay? And now the idea is, if I would like to, for example, predict the direction between SRC and PI3K, what I could do is check the proximity of the first node, this one, to the start of the pathway, and compare it to the proximity of the other node to the start of the pathway, and vice versa. Check the proximity of the first node to the ends of the pathway and compare it to the proximities of the other node to the ends of the pathway. And by designing a score that takes into account these mutual proximities, I can try to guess whether the first node is closer to the start and farther from the end, or vice versa, and therefore predict direction. And it turns out that this very simple approach is very effective. So on this specific pathway, but it generalized to other pathways, it gives very high accuracy, and as you can see, it's very, very simple. However, when my former student Daniel started looking at this method, he realized that we made a lot of arbitrary choices. For example, why would we score an edge by these multiplication operations and division operations? Maybe we should simply take all these proximities that we measured and use them as features for classification. The other thing he realized that, for example, when you check the proximity from a node to the ends of the pathway, there are many proximities that are involved there. For example, the ends could be some subset of nodes. So it's not clear why you should, for example, sum all those proximities together to get one measure. Maybe you should treat each proximity by itself, which means that now you get another set of features. But the problem is that all these sets have different sizes. It depends on the samples that you are using. And so we needed some component that will be able to learn from sets of different sizes. And luckily, there is a component that is called deep set, and it's based on the fact that you can learn a function on general sets by using this very a general decomposition in which you apply some function to each element of the set, you sum all these applications, and then you apply another function. And since we have functions here, you can approximate them using neural networks, which gives rise to the architecture or model that we came up with. And this model is, again, based on the idea that we take all these proximities of the nodes from the starts or from the ends of a sample, of a pathway. We input them to this deep set component, and we get one number, some set function that the model learns by itself that represents whether the edge should be directed in this way or in this way. So the overall scheme of the model we came up with, which is called DO, or Deep Orienter for short, is as follows. We take all the samples in the input. We also take a general network. And now for each of these samples, we measure these proximities of an edge to the starts or to the ends of the samples. I will soon say what do I mean by that. We get these distributions of proximities. We use the deep set component to transform them into numbers, and then we apply a last layer of multi-layer perspectron to generate the actual prediction of the direction. And so what are these starts and ends of a pathway? So in this, it's very simple, right? You have knockouts, you have effects, everything is very simple. In human, you have to take some surrogates of that. And the main surrogate that we used, which coincides with the focus of this meeting today, is cancer genomics data. So in cancer genomics data, in each sample, you have genes that are mutated. We think about them as causes. And you have genes that are differentially expressed. We think about them as effects. Okay? And so for each sample, we now have these causes and effects. And to say what does this sample believe about the direction of an interaction, we measure the proximity of one of the nodes, again, to the causes and effects. The other, we get this number, and we make the prediction. And so here you can see across different cancer genomics data sets, you can see the performance of our new method compared to the previous state of the art. And you can see that not only we surpass this state of the art, but we can do so with much fewer patients or with much fewer samples. So the difference is, in fact, much bigger the smaller data we have. And so just to show you one application of these directions, let me talk about prediction of cancer driver genes. And the idea here, so now we have a network with the extra information that we know the direction of each edge in the network. So the idea is that if we now diffuse information in the network, because of the directions, the information, the flow is limited, right? It is limited to follow only the directions of the edges. And so it cannot propagate too much. It's very focused. And so we believe that this gives us extra power. And indeed, what we did was as follows. We took different samples. We used only the sets of differentially expressed genes in those samples. We reversed all the directions in the network. And now we propagated information from these differentially expressed genes. And the idea was that all these differentially expressed genes were caused by cancer driver genes. And so if we reverse the direction and now we let information flow, we would hit those cancer driver genes. And so we checked this very simple idea using a comprehensive list of 930 known cancer driver genes, mainly from CGC. And these are the results that we got. So what you see here is the enrichment in the top predictions, in the top 0.1% predictions, up to 1% predictions, the enrichment of cancer driver genes in each of three methods. In gold is our method, in blue is the previous state of the art, and in green is when we are using an unoriented network that has no knowledge of directions. And again, you can see here that indeed orienting the network increased its predictive power. So to summarize, I've shown you two methods that we developed. The first for integrating multiple data sets in order to compute informative gene and pathway embeddings. And the second to orient a network to increase its predictive power. This is Rami and Daniel and my group, and thank you very much.
Thank you, Roded. I really find this inter or transdisciplinary research as fascinating. I'm puzzled about one thing that you said at the end, that it actually got you to have, that it was enough to have a much smaller size of data set, when I thought when you were speaking about replacing the formula from dividing it to multiplying, that it will actually lead to a need for much larger cohorts of data. Can you explain that?
I think in this case, the proof is in the pudding. There is no simple explanation. It's just that the method is more effective, and therefore it can learn better from smaller data compared to the...
That's impressive.
But that's a good question.
And another thing, at the end when you spoke, I'm wondering about causality and effect with the new compared to the standard that was before. Do you need or do you find more relevance to closer proximity between causes and effects in order to see the effect, or not necessarily? It's enough to look at distance as well from yours compared to, if you compare it to the standard that was before?
So it's a good question. I didn't look at the numbers, but in general, the network is quite dense. So, the average distance between every two nodes is maybe only 3 connections, but I didn't look at it specifically.
Okay, be interesting to see. Asaf?
Hi, wonderful talk. So you probably get this question a lot. Not all networks can be oriented, correct? So what happens if you try to integrate oriented and non-oriented networks?
Yeah, so I don't have the slide to show it, but I get this question a lot. And what we show is, as I said in the beginning, only about 2/3 of the interaction in the network are expected to be oriented. And when we check what interactions we cannot make predictions for because we get the same score for going this way and that way, then they are enriched in interactions that lie within protein complexes. So this is another validation of the method. I don't have the slide to show.
Ron?
Great talk. Did you, in this drivers analysis, did you try to go back and check what fraction of these putative drivers were actually mutated in those samples?
So can you repeat? I didn't get it.
You get the gene expression, you propagate the information back, and you get a putative driver. For this to be the real driver, the gene had to be mutated or somehow modified.
But the analysis somehow integrates many samples together in order to identify these drivers. So I guess, we would need to think about a subtle way in order to check this idea. And it might be also circular, right? Because
Okay, thank you, Roded. So with this, we end this fascinating day. I want to thank first and foremost Merck and Teva Pharmaceuticals for sponsoring this day for the 5th time. Hopeful to get next year again to see everybody here with your kind support. I'd like to thank Dr. Yudit Ben-Porat for being my partner in crime and in this really wonderful help. And of course, everyone, all the speakers from both sessions who on a personal note, gives me some hope, especially within this terrible period of time, that the future lies within science and the clinical translation, where it comes with huge responsibility to the future generations. So thank you, everyone. This was really a fascinating day. And I'd love to see you next year in the next World Cancer Day. And in the meanwhile, I would like to invite you to our Cancer Biology Research Center meeting in September. Todd Golub will be here as a plenary speaker, and I invite you to register. Thank you, everyone, and see you next year. Thank you.